Good morning, or I should say afternoon, everybody. Thank you for joining us here for Elkin Isaac. This is the 31st Annual Student Research Symposium for Albion. You're joining us here in Forum 4, which covers some biology and psychology presentations. We wanted to get started so that we let our first presenter begin right around at 1 o'clock. I would like to thank everybody again for joining us, and just a reminder that we will be recording these sessions. Please do not unmute your microphone during the session. If you have a comment or a question instead, please put those in the chat box and they will be read at the Q&A session at the end of the presentation. It's my pleasure to present Patrick Mayo, whose supervisor is Dr. Abigail Hill. And go ahead and start, Patrick.
Well, thank you for that introduction, and thank you all for coming. As she already said, I'm Patrick Mayo, and my advisor is Dr. Abigail Cahill. And today, my presentation will be about the ecological impact of a genetic superclone. Next slide, please. So, the species of interest that I'll be discussing today is Aphis neri, or the oleander aphid. This aphid, as the name would suggest, feeds upon the plant oleander in a pest-host sort of relationship. So unfortunately, while being a pest to a plant is already bad enough, this invasive insect has made the jump from a plant that some may be familiar with to a much more familiar plant, milkweed. You'll know this as the host plant to monarch butterflies and seen pretty much all over Albion and in this area of the world. So the thing to note with this invasive species is that they have an incredibly low genetic diversity. This is because how they reproduce comes in two different ways. Primarily, the females just deposit nymphs that are already rip-roaring and ready to go, and the other is sometimes they have sexual reproduction. This typically is not the case, so effectively they can clone themselves with very little genetic diversity from the females. Next slide, please. As you can see here, this is the species of interest. They look pretty distinct with their orangish yellow pattern, and you may be familiar with most other aphids being smaller and a little bit more green. These are definitely more distinct, and if you ever catch yourself near some milkweed on campus during the summer, you'll definitely see quite a few of these guys. Next slide, please. So, onto the project. The project was divided into two separate parts, an ecological study and a genetic study. The aim of the study in general was to collect data in the hopes of determining patterns of distribution for the oleander aphids living in this part of the United States. So in terms of the ecological study, the data was collected in September of 2019 with a collection range from here in Albion to over into Reading nearby, Michigan, and into northern Indiana, specifically Auburn, Indiana. The data that was collected in the collection were measurements of the milkweed plants, and those measurements included the height of the plant, the number of leaves, the population density of other milkweed plants in the area, and more importantly, we took measurements of the aphids and how they were distributed on these milkweed plants, where they were located, how many there were, et cetera, et cetera. So we ended up collecting 3 different samples of these aphids at Albion and Auburn, Indiana. There were 2 in the Albion area that we took collections from. Next slide, please. So this graph represents the distribution of aphids on milkweed relative to how tall the plant was. Something to note with this graph and data, it does not include the height of the milkweed plants that had no aphids. So what you're seeing now are all the plants that had aphids on them and the amount of aphids there were. So while The data appears to have a negative correlation with the aphids seemingly to prefer shorter plants. This is not the case. The data isn't statistically significant, and as you can see, you have quite a few outliers that sort of break this apart. Next slide, please. What we have here is a similar graph. The graph that you see here represents the distribution of aphids on milkweed relative to the number of leaves on the host plant. And again, just like with the previous graph, this graph does not include the milkweed plants with no aphids. So what you're seeing is just plants that have aphids and their number of leaves. You see a bit more of a stronger correlation, but again, it's not statistically significant. We have outliers both above and below the line. So next slide, please. So what's the conclusion from the ecological study? Again, we didn't really have much of a correlation. We have some patterns that would be interesting to further investigate. But as of right now, we couldn't really determine a strong pattern with the data that we took. Next slide, please. On to the genetic study. So, as mentioned previously, samples of high density of the population were taken at 3 different locations, 2 in Albion and 1 in Auburn, Indiana. So, with these samples we took, the aphids were individually separated. Their DNA was extracted. PCRs were performed on that extracted DNA. And then those PCR products were sent off to U of M, the University of Michigan, for sequencing to better see how closely related these are, the genetic difference, so to speak. Next slide. So what did we determine? Well, unfortunately, the sequences that we received back from U of M didn't really yield any usable results. It's worth noting that one of the sequences actually had a high similarity of up to 50%, but the matching species that it was associated with were very different from the aphids that we were studying. They weren't even really in the same family. It was bivalves, some mollusks. Yeah, it wasn't really usable data, unfortunately. So, as with the ecological studies, more data is definitely needed. And another attempt, I think, is needed to collect samples and extract the DNA, hopefully with a bit more usable results. Next slide, please. So, what does it all mean? So, the study provided a lot more questions than answers. I'm not sure if that makes it a good study or a bad study, but it was definitely an interesting one. So some of the questions that we sort of walked away with are, did the aphids have a strong negative effect on the milkweed plants with that pest-host relationship, while the pattern was seemingly that they preferred shorter milkweed plants with less leaves? We're wondering did they cause that, or was that just a matter of their selection? So something else that looking forward, other experiments and studies worth noting would be when the aphids actually showed up, how long do they remain, how quickly do they reproduce, and I think we have a pretty good outline for a future experiment as far as determining when they arrive, how often they reproduce, and what effect will they actually have on the host milkweed plant of interest. We're also looking into how they're actually being dispersed throughout and where they gather, where they congregate, and if we can determine a pattern of where this invasive species is going to leave a larger impact versus a smaller impact. Next slide, please. All right, before we get to the Q&A, just want to give my resources and acknowledgments. This is what I have for the resources. Next slide. And here are all my acknowledgements. I'd like to thank the Elkin College Biology Department, obviously, the U of M Science Department for the sequencing. As always, Dr. Abigail Cahill for being a great mentor and advisor. In the samples that I took, I went up north to see my grandmother, and she happened to have some milkweed around. And just as well, in Indiana, I knew a few people, and they were kind enough to let me wander on their property with a meter stick and a pad and paper. So just like to give them a thank you. All right, on to Q&A.
All right, so just as a reminder for anybody who has joined us since the beginning, if you have questions and can post them in the chat, that would be great. We have our first question from Doug White, asking if milkweed populations are in decline, could aphids have difficulty tracking the plants?
Good question. I'd have to imagine so. We don't necessarily know so much on how they distribute themselves and actually go to milkweed versus oleander, but I'd be interested to see if we could actually model an experiment to see if they prefer areas of low density milkweed versus high density milkweed. Thank you for that question.
I will ask a question while we're waiting for any others to come in. You mentioned a couple of times with your graph, your two graphs, where you looked at the height of the plant and the number of aphids on it, that you had only considered plants where aphids were present. Did you look at, or did you have any sense for whether there was a pattern in terms of what the characteristics were of milkweed that didn't have aphids versus did?
Good question. So from essentially separating the data into those two different parts, the milkweed plants that had aphids versus the milkweed plants that didn't, couldn't really see much of a correlation. Thus, aside from the fact that most of the milkweed plants that didn't have aphids, they seem to be a little bit healthier, so to speak. Health just being the coloration of the leaves and the height of the leaves. But I'd be interested to actually redo that experiment and look at those milkweed populations to make note of those characteristics. That may actually reveal a more interesting pattern, so to speak.
Any other questions for the chat? I have another question that's just come in that says, are there specific methods for removing the invasive species or protecting the plant from them? How do these methods impact the native species or plant?
Interesting question. This isn't exactly my area of expertise, but I would say if you have a large population of milkweed and you want to prevent pests from getting on it, you would use pesticides, so to speak. And I'm sure we can imagine that those have a lot of negative downstream effects. As far as I know, milkweed populations aren't really that protected, ecologically speaking. They tend to just grow plentifully enough. But if those populations get smaller, then it may be worth looking into to see what ways we can sort of deter these pests from the milkweeds and actually allow them to be utilized by the monarchs and all that.
Are there any other questions? All right, I have one more that may be a comment. This is, I know that in this job I had in the forest preserve, there was a brush fire that got rid of them after we would cut them, and they would put down pesticides. That's from Michaela Hawkins.
That may not actually be such a bad idea, especially if you can reconstitute the milkweed plants somewhere else. Interesting. Thank you.
And I have one additional question from Shanti, asking how would you convince a friend or family member that the milkweed is important and not just a weed to be removed?
That's a good gardening question. Well, you want more diversity in your garden, obviously, so you can attract more things to have it be more pleasant to look at. Milkweed already is kind of a very pleasant plant to look at. It has nice curves, nice lines. Just as well, that milkweed is actually quite vital for the monarchs, and obviously monarchs are very lovely to look at and behold. So you'd have to appeal to their nature in the artistic sense. I would suggest you start there and then work your way into this is actually good for the ecology of your environment.
All right, it looks like we may be done with questions and we're about at time. Is that correct? Okay.
That's correct. Thank you,
Thank you very much.
Thank
Nice
you.
job. All right. Thank you, everyone, for joining us. For those of you who are just joining us since the last presentation, welcome to the Elkin Isaacs Student Research Symposium. Just a reminder, we will be recording this session. And also, please do not unmute your microphone to speak. Rather, if you have a question or comment, put it in the chat, and we will ask that to the presenter at the Q&A session at the end. It is my pleasure to present Audrey Lewis, whose faculty sponsor is Dr. Skeen.
Thank you. So my presentation is about my first project that I did 2 summers ago, which is a taxonomic revision of the Myconia impressionibus complex on Hispaniola. Essentially, it's a project about botany. Next slide, please. So to start off, a taxonomic revision is a study that essentially compares populations of organisms to figure out how they should be classified. And these kinds of studies are valuable because it helps you understand the evolutionary relationships between organisms and their close relatives. And it offers a sense of predictability in a way. For example, if there's a newly introduced species, could it possibly become invasive? And then if a new drug or gene is discovered for an organism, is it possible to look for it elsewhere? Next slide, please. So I studied shrubs and small trees in the melastome family, which is called Melastomataceae. And melastomes are typically characterized by their opposite leaves with arched longitudinal veins and nearly perpendicular cross veins, as well as the fact that they have well-developed hypanthium, which that just means they have a fused base of sepals, petals, and stamens on the flowers. And there are about 4,200 to 4,500 named species, and they're primarily tropical. And they're separated into different groups that are called tribes. Next slide, please. This is a picture where you can see the arched longitudinal veins on the leaves, and Dr. Skeen took this photo. Next slide, please. The tribe Myconii of Melastomataceae has about 1,900 species separated into 17 different genera, and about 1,000 of those are in Myconia. The National Science Foundation did fund a planetary biodiversity inventory grant to help study this evolution and the diversification of the group. And the grant was centered at the New York Botanical Garden, as well as the University of Florida, the California Academy of Sciences, and a university in Brazil. Next slide, please. And the tribe Myconii has long been known in the Myconii, it's been known that the genera in the Myconii were arbitrary. And the PBI grant resulted in all 17 genera in Myconii being just lumped into a single monophyletic genus called Myconia, and of about 1,900 species that are defined by berry fruits and also tiny leaves, which are called bracts, under each flower. Next slide, please. So the old name for Myconia was Chalcogonium. So if you look at old names for these species, that's why you'll see Chalcogonium. And so this was a genus of about 40 named species, and they're polyphyletic, so they have multiple clades or evolutionary lineages, and they are endemic to the Greater Antilles. Next slide, please. So this is a photo of a ruptured calyptra, and the reason why I'm showing this is because 3 of the main defining features of the species in Myconia, they have a calyptra, which is essentially a protective covering over the young flower bud that has not yet emerged, and they have mite domatia, and they're glabrous, which means they're hairless. So this photo, you can see where the young flower bud, the petals, they've pushed through and they've broken that calyptra open. Next slide, please. So this photo on the left, you can see that the calyptra has been destroyed by petals that have emerged through. And on the right, you can see the mite domatia in the junctions of the veins. Next slide, please. So my objectives during my study, I wanted to define what species were in the species complex on the island of Hispaniola, which is Haiti and the Dominican Republic together. And tentatively, there are 4. So you have Myconia berdiana, Myconia ericmaniana, Myconia impressonervis, and Myconia thorbeckiana. And these 4 species are very closely related. So any botanist that decides to study them can easily be confused. And then another one of my objectives was to write descriptions of the species as well as an identification key. Next slide, please. So during my study, I mostly worked with dried herbarium specimens as well as online images of herbarium specimens, and the data that I collected from them allowed me to determine what species there were. So I looked for gaps in morphology as well as I looked at the geographical distribution differences as well as the habitat differences. Next slide, please. So this is Myconia berdiana, and this is a picture of an herbarium specimen of it. And then there's a map where you can see that it has been found to grow in the Dominican Republic. Next slide, please. And this is Myconia ericmaniana, the herbarium specimen photo, and then the map of where it's been found in Haiti. The little dot is all the way on the left. Next slide, please. And then Myconia impressonervis, the herbarium specimen photo, and then it's been found to grow in the Dominican Republic. Next slide, please. And then you have Myconia thorbeckiana, the herbarium specimen photo, and it's been found to grow in Haiti, as shown in the map. Next slide, please. So for my methods, I compiled the label data from the herbarium specimens, like I said, looking at the specific geographic distributions and the habitats, as well as the phenology, so the flowering and fruiting times of the plant. And for measuring the specimens, I used a 15-centimeter rule and then also a tiny measuring scale to use with the microscope. And so I used a dissecting microscope with the appropriate camera to pair with it in the software. And then for the digital herbarium specimens that I looked at on my computer, I used a program called Xscope for the Mac operating system, and then IC Measure for a Windows operating system. Next slide, please. This is a little action shot of me using that dissecting microscope. Next slide, please. And then this is what I would see when I would be using Xscope on my computer to measure some of the leaves. Next slide, please. So for my results, essentially, I became more familiar with the microscope and the software, and I was able to capture images of the plants, and I measured the flowers and leaves, and I was able to write a basic taxonomic treatment with an identification key. Next slide, please. So this is the taxonomic treatment. I broke it into two different slides so you could see. So essentially, it's a series of choices that you follow through and you read through each one. And if the description matches what you're dealing with for the plant you're looking at, then you can just follow through and it'll help you figure out what plant you're studying. So this is the first half. And then next slide, please. This is the second half for it. Next slide, please. And I would like to thank Dr. Dan Skeen for making this project possible, as well as the Albion College Biology Department, and Renee and Dr. McCaffrey and the FIRSCA committee. Next slide, please. And some sources that I used. Next slide, please. And then any questions?
Thank you, Audrey. As a reminder for anyone who's just joined us, if you have questions for Audrey, we would love it if you could put them in the chat. So our first question is from Nathan Cruz, and he's asked, does Myconia reproduce sexually or asexually, and is that a trait because it is found on more warm islands?
That's a really good question, and I'm actually not sure about that. I never looked into that, but that's something that I will investigate for sure. Thank you for that.
I will ask a question while we're waiting for more to come in. What did you find to be the most challenging part of trying to tell these species apart?
I would say that because they're so closely related, a lot of the physical features closely resemble one another. So really just paying attention to the measurements and like any minute difference you might find, you really have to take into account. And that was definitely taxing at times because it was very monotonous. But yeah, I would say that would be it.
Our next question is, where do you get the dried specimens? What are the benefits or drawbacks of using dried specimens or pictures?
Good question. So the dried specimens I got from a couple of different herbariums. So some were actually from the Elkin College herbarium, and then some were, for example, from the Missouri Botanical Garden and a couple others. I would say there weren't really any drawbacks of using the dried specimens because you had it physically in front of you and it was easier to measure and just to see in person. But the pictures were difficult because you had to make sure you were measuring to scale. Like depending on how zoomed in you were on your computer screen, you had to make sure your ruler that you were using on your operating system was at that scale as well. And if you didn't make sure of that, you would have to go back and like redo them. So that happened to me a few times, which was a drawback for sure.
Any other questions for Audrey? We have a question from Patrick Mayo asking, what do you intend on studying next?
Good question, Patrick. At this moment, I don't intend on studying anything next, but I will be interested if anything does pick up with the study, possibly with Dr. Skeen in the future. I would probably jump back into it, but as for now, not really anything.
We have a question from Nathan Perriat asking, what would be the next step if there are any for this research project?
That's a good question. A next step would be potentially incorporating this research and doing some more and possibly contributing to a publication that Dr. Skeen could be working on, but that would be about it.
We have a comment from Jill Marie Mason saying, great work, Audrey. Thanks for participating. And then a question from Shanti asking, why is it important to define the species in detail?
First, thank you, Jill, for that. I would say it's important to define the species because when you're able to define a species, you're contributing to the overall understanding of biodiversity and the overall understanding with Earth. So I would say that would be the main importance of that. And then also helping other botanists that choose to study the species or maybe a closely related group, it would just help them become less confused since we have more information on the species.
And a question from Doug White asking, are there conservation concerns for these species?
I do believe there are some concerns because I believe there is an issue in Haiti with deforestation, which could potentially affect these plants if they're growing in an area where that is a problem. But to my knowledge, aside from that, I am not 100% sure, but that is something I will look into. Thank you.
And a question from Sheila Lyons-Sabaski asking, what was your impression of the software used to measure the plant characters? Was it easy to use? How could it be improved?
My impression of the software overall was good. And if I had to pick one to use for the entire time, it would have been the iMeasure, the IC measure with the Windows operating system. It was a little more user friendly in regards to making sure it was set to scale. I would make a couple of changes to the Mac operating system on the Xscope just because of the scaling problem and how it was measuring in pixels. So you had to make sure the number of pixels matched up with what your ruler was. But that would be it.
Thank you, Audrey, for your presentation. Thank you all for your wonderful questions. We are going to move on to our next presentation. For those of you who are joining us, welcome to the Elkin Isaac Student Research Symposium. Just a reminder, we will be recording the session, and please do not unmute your microphones, but if you have a question or a comment, please put it into the chat as we will read those during the Q&A after the presentation. It's my pleasure to introduce Nathan Perriott, and his faculty supervisor is Dr. White. Go ahead, Nathan.
Thank you. So as she said, I'm Nathan Perriott, and my faculty advisor was Dr. White, and we looked at the effect that parasitic mites have on incubating house wrens. Next slide, please. So some background information on the house wren. It is one of the most common birds in the Western Hemisphere, especially in the White House Nature Center. It is a cavity nester. Its primarily food is insects. And while nesting, it prefers very high temperatures while incubating its eggs. And compared to a human, there's a huge difference in temperature, as the house wren tends to keep its body temperature around 42 degrees Celsius compared to a human body, which is around 37 degrees Celsius. Next slide, please. So the mites that were evaluated in this study were feather mites and red blood mites, which the red blood mite is the picture around the screen. So the feather mites eat away at the keratin and feathers of the birds. And for those who don't know what keratin is, that's the main structural component of the feathers. And as for the red blood mites, they were mostly active at night and in the early morning when they are feasting on the bloods of the bird, and in this case, the house wren. Next slide, please. So for the incubating patterns, I hypothesize that there is a correlation between the amounts of mites in a nesting box and how long the house wren will incubate her clutch. And this is basically studying whether or not the amounts of mites in a nest box will determine how long a female house wren sits or is away from her nest on top of her eggs. And as for the null hypothesis, that we've predicted that the number of mites in the nest will not change these incubating patterns of the house wren. Next slide, please. So for a starting point, Dr. White had some old iButton records from previous years, and these iButtons kept the temperatures from the bottom of the nest box, the lid of the nest box, and in the cup, which is the nest of the house wren. These record the intervals of temperatures in degrees Celsius in minutes. These also converted into graphs, which were analyzed later. And these graphs were analyzed minute by minute to record on and off bouts. Now, an on bout is considered how long the female house wren was sitting on her nest, and an off bout is how long away the house wren was either off her nest or off foraging. And as you can see from the picture, the silver eye button compared to the eggs, the female house wren would just sit on it like a normal egg. Next slide, please. So for our methods, the female house wrens must constantly balance time on and off the nest to ensure the growth of her clutch. So for determining the starting point of the graph, as this picture, the flat line in the beginning of the graph will be considered the overnight that the female house wren was sitting on her nest. And from where the flat line ends to the first dip in the graph, that's considered the first off bout, or the first time period that the house wren is away or out of her nest box. And then the first on bout would be considered from the low point of the graph to the first peak. And that would be how long the female house wren is back on her nest. And we recorded that 5 times. Next slide, please. As for the mite levels, we studied 4 levels of mites, which went from none to low to moderate to high. In the top right, we have 0 mites on the nest box. Those were the ideal nest for the female house wrens. In the top left, we have a low level of mites, which they are really hard to see because they're small, but there's not many there. The bottom left was considered a moderate or a level 2, where there's considerable amounts of mites. Then the bottom right is level 3, which is not desirable by the female house wrens. It could cause significant problems. Next slide, please. So for an example of the data we would analyze, we would take the eye button records from one nesting box, and we would analyze this data for 10 days. We record the first time the house wren left to nest, which generally would be at sunbreak. We'd record the temperature inside the nest and outside the nest to compare if there might be any correlation between daytime temperature and nesting habits. Then we record the on and off bouts. We recorded 5 off bouts and 5 on bouts. At the end of each of those bouts, we also looked at the temperature of the lid inside the nest box and the bottom of the nest box to evaluate how warm the nest might get between the on and off bouts compared to when they first left the nest overnight. Next slide, please. So for the individual level results, the nests with no mites tended to sweep in longer, which means they typically left the nest at a later time in the morning compared to the severe mite level nest boxes, which tended to leave earlier. The nests with no mites tend to have the highest temperature in the morning, which is preferred by the house wrens. And this is because the female house wren gets going back to the sweeping in longer. She gets to stay on top of her nest longer, which is preferred. And the more mites in the nest box was the longer the female house wren spends on and off the cup. She probably spent more time on the cup, picking away at the excessive amounts of mites, and she spent more time away from the cup because she needed to find more food and forage to keep up her energy that she is spending trying to get rid of the mites. Next slide, please. So then I grouped the nest mite levels together to see if there were any other significant changes in results. I grouped the level 0 and 1 mite nest boxes, which were none to low, and levels 2 and 3, which were moderate to high. When grouped together, there was really no change. Level 0 and 1 also tended to sweep in longer. There was, on average, about a 5-minute difference between those two nest boxes. And they also both spent shorter intervals spent on and off the cup, which were relatively consistent time-wise compared to each other. Levels 2 and 3 typically left the nest earlier, which would be on average 7 to 10 minutes before the level 0 and 1 house wrens would leave their nest box. They also together also spent more time on and off the cup. Next slide, please. Interpreting these data points on a large scale, a t-test failed to identify large differences between the different years of mites. If I were to continue on with this study, I'd like to, or if somebody after me, since I'm graduating, I'd love to see more previous years compared to the 2017, 2018, and 2019 data graphs that I looked at. According to the t-test, there are no large correlations between the mites and incubating house wrens. And as I said, reviewing and analyzing data from other previous years could yield more patterns. I analyzed the on and off bouts, but I'm sure the temperature, since we only recorded the 2 data points of temperature, I'm sure there could be more patterns to be looked at. Next slide, please. For the acknowledgements, I'd like to acknowledge Dr. White and Dr. Kennedy for both expanding my knowledge in the ornithology field and for this project. I'd like to thank the FIRSCA committee, Renee and Dr. McCaffrey, and the Irwin Fellowship for also funding this experiment. Next slide, please. And here are some literature cited that I used over the course of my study.
All right. Thank you, Nathan. So we will move on to the Q&A portion of the talk. So if anyone has questions for Nathan, if you could post them in the chat, I will go ahead and read those off to him. And while you're doing that, I'll start with a question. Do you have any ideas about what might lead to variation in which nest boxes have lots and lots of mites and which don't have very many?
Previous years, Dr. White and Dr. Kendi, they go through and clean some of the nest boxes. Unfortunately, some nest boxes just tend to have more mites after they are cleaned. But I do know they can, Dr. Kendi and Dr. White also help control the damage and the amounts of mites in each nest box.
Thank you. Our next question is from Jeff Perriott asking, what p-value did you use for t-test failure criteria?
I used the two-tailed hypothesis or two-tailed t-test hypothesis test, and we did a p-value of 0.05.
We have a question from Audrey Lewis asking, what was the most interesting part of this project for you?
The most interesting part is that this was not the research project I intended to do as COVID impact. I was supposed to be in the field over the summer here at Elkin, and I was actually going to be in person, like controlling the mite levels and stuff. And then once we all got sent home, Dr. White was able to come up with something in a pinch, and that was just analyzing the data from previous years to find correlations and patterns.
And we have a question from Shanti asking, why do the wrens prefer certain temperatures?
Higher temperatures are preferred for the eggs during incubation. It helps with development of the birds inside the eggs. And if temperatures get too low or too high, they might die. But a higher temperature than the human body allows for just this certain type of perfect growth for the house wrens inside the eggs.
Other questions for Nathan? We have a question from Patrick Mayo. First said, well done. And then also asked, do you think there is a relationship between clutch size and mite population density?
So on the grass, we recorded the clutch sizes along with the mite levels. And this, going back to what I said about maybe future studies, that is one thing that we have the data to look at to see if the mite levels affected how many eggs that the house wren laid. And we could also look at how many of these fledglings also survived or died. But that is something I did not look at during this study. But it could be done if somebody was to like continue on this project. I have all the data for it.
Our next question is from Nathan Cruz, who asks, mites prey on the wrens who have a higher body temperature. Are mites specifically attracted to the heat of their prey?
As far as I'm concerned, the mites are just attracted to the birds. I didn't really study into the specifics. I just kind of looked into the blood mites feasted on the blood of the birds. I'm sure the higher temperatures of the nest trigger the mites to be more active, because if it was cold, the mites would probably be more dormant. So I guess in hindsight, the temperature does make the mites more active.
And Dale Kennedy asks, what are you doing next?
I will not be pursuing a career in the ornithology field, unfortunately. I will be attending graduate school somewhere in the near future and then medical school after that.
And then I think we have probably time for one more question from Abigail Cahill. Do you think there's multiple species of mites in these boxes? Might that explain some of the variation?
I do not think the specific types of mites would cause a variation. I think it's just the mite levels of the nest boxes themselves. I would assume a low level amounts of mites aren't going to cause much problems at all. It's just the more mites there are in the nest box, the more problems there's going to be.
Thank you so much, Nathan, for your presentation. Hello, everyone. Welcome to those of you who might be joining us. Just a reminder, we are recording this session. And also, we would ask that you not unmute to ask any questions or make comments, but rather put those questions and comments in the chat box, and we will ask them during the Q&A session following the presentation. It is my pleasure to introduce Irene Corona Avila and her project working with faculty supervisor, Dr. Cahill.
Thank you for that introduction. As mentioned, my name is Irene Corona Avila. My advisor is Dr. Abigail Cahill, and I'll be explaining my research investigating genetic diversity of Orlander aphids in the United States. Next slide, please. And so the species of interest is the Aphis nuri. They're also known as the Orlander aphid, and you'll hear me refer to them as aphids. They are Mediterranean native, and they inhabit different regions throughout the U.S. These aphids are invasive to the milkweed plants. It's important to know that when we're looking at these aphids. And this is a continuation of Katie Ferrero's investigating aphid superclone status study that she did in 2019. So it's been a few years since we looked at the genetic diversity of aphids. Next slide, please. And so what is the purpose of this study? Why is this important? Monarch butterflies habitat and feed off of milkweed plants, but so do the invasive species of aphids. And we may be looking at a correlation between aphids harming milkweed plants and therefore contributing to the decline in monarch butterflies, though there's still little evidence to support this hypothesis. And so what we want to do to understand aphids, we're going to look at the reproduction and recolonization. I want to note that female aphids reproduce asexually during the summertime, and they die off in the winter. And in a 2011 study, Harrison and Mondor found aphids from Georgia and California that exhibit multilocus genotype, which means that they exhibited low genetic diversity. So this was really interesting to see, because as aphids recolonize to different regions, we would kind of expect those aphids to adapt to different environmental conditions. And so the purpose of this study is to look at the genetic variation by geographical recolonization and isolation. In doing this, I proposed two questions. The first being, where do aphids recolonize? And when I mean by recolonization, I speak about the different areas where aphids habitat an ecological niche by a single population. So for example, did aphids in Michigan come from Tennessee? Did they come from Georgia? Or did they reproduce in their native region? And with answering this question, we can start building off a map of aphid recolonization within the US regions to further understand this. Second question I want to answer is, how genetically diverse are these aphids? And based off the 2011 study, we expect low genetic diversity. Next slide, please. So we collected aphids from Albion, Detroit, Sault Ste. Marie, South Haven, and Pennsylvania. As I mentioned before, I am continuing Katie's study in 2019, and she only had 2 populations from Michigan and 1 from Pennsylvania. So this is just expanding the library, the data that we have on aphids. And so I extracted the DNA, and once I successfully extracted DNA samples, and as you can see to the right, this figure is a gel from DNA samples of Detroit. They're beautiful bands, you can see them. And so I was very excited when we got these bands, because as many scientists know, running a gel is so difficult. And once we've received successful DNA samples, we ran a PCR through a CO1 gene. And the reason why we used the barcode CO1 gene is because it's really easy to work with and it had a broad reference database. And when received successful PCR samples, we sent them to the MSU Genomics Core for sequencing. Next slide, please. And after receiving the data from the MSU Genomics Core, we ran them through the R programming, and we got this phylogenetic display that you see on the screen that shows the genetic relation between aphids. And so a phylogenetic tree is a branching diagram that reflects how species evolved from a common ancestor. In other words, the distance between two branches show how closely related those two species are. And as you can see, we have aphids from Michigan, South Haven, Sault Ste. Marie, and Albion that are kind of closely related to each other. And then you see this odd one from Albion that's at the top of the phylogenetic tree, right below Pennsylvania and separated from other Michigan aphids by Pennsylvania aphids. And so this is interesting because we would expect to see Michigan aphids closer to each other since they come from the same region, but we see that they are more related to Pennsylvania aphids. And when we're looking at this, you see that South Haven also has a branch in between Pennsylvania aphids, but they're still kind of closer to Albion and South Haven and Sault Ste. Marie. Next slide, please. And we also did a pie chart displaying the genetic relation between aphids. And so this pie chart, in understanding this, the circles size is proportional to the number of individuals with that sequence. And the further apart the circles are, the more different the sequences are. The circles in red are aphids from Pennsylvania. The circles in blue are from Albion. The green are from South Haven. Purple are from Sault Ste. Marie. I did mention earlier that we did collect aphids from Detroit, but we found that they were infested by these parasitic wasps, which was really interesting to find. So these parasitic wasps, what they do is they use aphids to host their babies. And so I found that this was really interesting that we only found it in the Detroit aphids. And so the large circle of aphids, the red large circle, shows that there were multiple aphids from Pennsylvania with the same genotype. And so you see that there are still smaller red circles that signify Pennsylvania aphids, but they just show that there's just one small little thing that makes them different from that other Pennsylvania aphid. And just like the phylogenetic tree displays, the Sault Ste. Marie and the Pennsylvania aphids are least closely related than they are to other aphids in Michigan. And so this shows that we meet our expectations given the clonal ecology that aphids express low genetic diversity. So it may seem like these aphids are far different from each other, but this is just like a zoomed in view of the phylogenetic tree. And they still are closely related to each other, but there's just one small thing that makes them different. Next slide, please. And so in conclusion, where do these aphids recolonize? So we suspect recolonization of aphids between aphid and Pennsylvania regions. We think that they may be coming from the same region, maybe from the south, as we expect like aphids do not live in the winter. So we believe that they may be coming from the south, from the same region. And there may be a slight chance that South Haven may be coming from the same source as Albion and Pennsylvania aphids, since they were still in between each other in the phylogenetic tree. And second question, how genetic diverse are aphids between regions? As we expected, they showed low genetic diversity. That's really good. Next slide, please. So where would I like to take this? So I'd like to continue building off this map of recolonization. I think to understand recolonization, I'd like to collect and extract DNA from different aphids throughout the region. Currently, we have some aphids from Georgia that I collected over the summer, and I'd like to see if these have the same genotype as aphids in Pennsylvania or Michigan, and that may give a better sense of where these aphids are coming from. And so as you heard from Patrick, if you went to Patrick's talk earlier today, he saw the ecological relationship between aphids and monarch butterflies. And so I was more on the molecular side of it, but we saw a little evidence that there's milkweed plant impact from aphids because of the few caterpillars that we saw. Next slide, please. And I'd like to acknowledge Dr. Abigail Cahill for endless support and advising. I really appreciate her working me through this project. The Cahill lab, my lab teammates are very helpful and supportive. I'd like to also thank Katie for allowing me to continue her study, and the MSU Genomics Corps for sequencing the aphid DNA samples, and the Student Research Partnership Program and the Albion College Biology Department for funding and support. Next slide, please. Thank you everyone for coming on this beautiful afternoon. I hope you enjoyed the presentation. Does anyone have any questions?
This is just a reminder. If you have questions for Irene, if you could post them in the chat, I will read them off to her so that she can respond. All right, so our first question is from Nathan Cruz asking, did the parasitic wasps cause the genetic makeup of the aphids to change majorly, or was the data still usable?
So yes, it actually impacted a lot. We cannot use the Detroit aphids anymore because they were infested by these parasitic wasps. And when we received the back data, they consisted of almost 90% of the aphid DNA. And so we could not use the Detroit aphids. It was super sad, but we'll hope to collect more samples.
And our next question is from Patrick Mayo. First says, great presentation, Irene. And then roughly how many individual aphids did you sequence?
So that number has to be, I am not positive about how many aphids, but it has to be close to over 120 aphids that we sequenced. But not all of them came back with great results. So we were not able to use all of it. So that's why I hope to continue to expand the sample size in future research.
Our next question is from Jessica Kane asking, did you find the wasps in any other aphids besides the Detroit ones?
No, so we actually just found them in the Detroit aphids, which is really interesting. And I wonder if that was just like a regional thing, or maybe we may see them later because they do use aphids as a host. And so we may see that more in the future when we collect more samples of aphids.
We have a question from Trissandra Ragsdale asking, what areas would you like to collect from next? And also she says, great job, Irene.
Thank you. I would like to continue to collect aphids from Georgia, and I will be in Chicago this summer for a research internship, and I'd like to see if I can collect any aphids from Chicago as well. Maybe venture out in Illinois and see if I can get any samples from there.
We have a question from Bree Ruger asking, do all the aphids die off over the winter? Aphids are not the best flyers, so is there another way they could be arriving in the northern US from the south, or could they be surviving here somehow?
Yes, so the way that aphid reproduction works is female aphids reproduce asexually in the summertime, and when it comes to the wintertime, they start to die off because they can't handle the temperature, environmental conditions. And so there's a female aphid and a male aphid that reproduce and lay an egg in the wintertime. And when that egg hatches in time for the summer, then they start to reproduce multiple female aphids. And so we're thinking that the best environmental conditions that the best environmental conditions that these aphids can continue to, the aphid egg can continue to survive are in warmer temperatures. So we're thinking that these eggs that are hatching, they're coming from the south and then reproducing and recolonizing going all the way up north.
We probably have time for one last question, if there is one. All right, thank you. Thank you, Irene.
Thank you everyone for the questions. I appreciate you guys coming to this talk.
My apologies. Can you hear me?
Yes.
Okay, sorry about that. It said my mic wasn't working, so let me represent the screen. Are you able to see the presentation now? Okay. Thank you all for your patience. Thank you, Irene, for your great presentation. We are going to move on. Just a reminder for those of you who are joining us, thank you for coming to this presentation as part of Elkin Isaac, the Student Research Symposium. Just a reminder, we will be recording this session, and we would ask that you do not unmute your microphone. Instead, if you have a question or a comment, please put it in the chat, and we will ask the presenter at the end of their presentation. It's my pleasure to introduce Rosemary Hernandez, whose faculty sponsor is Dr. Cahill. Rosemary, are you here?
Try, so there, it did look like, Shanti, it did look like there was one that was already pre-recorded, so yes. Yep.
Thank you. I'm sorry about that. Let me make sure the sound is gonna work.
Good afternoon, everyone. Thank you for attending my online presentation. My name is Rosemary Hernandez. My faculty sponsor is Dr. Abigail Cahill, and my presentation will discuss phylogenetics and the distribution of Culicoides larvae in a Michigan inland salt marsh. So there's a rare inland salt marsh located in the Maple River Staking area, visually located on the map on the right. The area contains the only known salt marsh in Michigan. The marsh was formed from a pre-existing ocean during the Silurian or Devonian period. The ocean left salt rock deposits into the water and land, which then after a few decades created a natural brine aquifer. The brine consists of sodium and chloride dominant groundwater, which creates a mineral soil saturated wetland. The inland salt marshes are near streams or rivers, in this case a river, where glacial drift is thin enough to permit the brine to emerge seasonally. So since the marsh contains mineral-rich saline conditions, it causes a limited number of plants and invertebrates to tolerate the salt marsh. In this case, one of the invertebrates that is found in the marsh is the Culicoides biting midge larva. Biting midges are flies in order Diptera that as of now contain more than 4,000 species and are divided into many subgenera. The biting midges undergo metamorphosis, so the eggs are laid on moist saturated surfaces like the marsh and hatch in 2 to 7 days. The 4 larval stages are then completed in about 2 weeks to a year and finished in late winter. And the pupal stage is in the same site as the larval stage and fully formed in the spring. Adults emerge 2 to 3 days after in early summer. In the marsh, the Culicoides are found in their larval stage. In North America, there are 2 major species of biting midges, the Culicoides sonorensis and the Culicoides veripennis. Culicoides veripennis is a part of a subgenus Monoculicoides. So there are Culicoides veripennis complex flies, which are biting midges that were genetically similar to the veripennis, but contain a few mutations that made it similar to the other species under the same subgenus. So these biting midges are categorized as variants or complexes of the Culicoides veripennis. The Culicoides sonorensis is actually a subspecies of the Culicoides veripennis. So these biting midges are geographically isolated. So these 2 species live on the opposite sides of the US as shown on the map. So as you can see, the Culicoides sonorensis live on the west and southwest region, and typically found along the West Pacific coast. And the Culicoides varipennis live on the Midwest region, typically found in Illinois and Wisconsin, and have been rarely found in Michigan. So since the Maple River Staking Area contains the only salt marsh in Michigan, and there have not been many research studies on the various invertebrates that live in the marsh, I wanted to study the Culicoides specifically. I wanted to know what species the invertebrates was and where they are distributed along the marsh. So Dr. Cahill, the research team, and I went out to the salt marsh in 2018 and collected sediment samples along the transect that started from the salt seep to the freshwater area. We separated the transect into 7 points, 10 meters apart. The first point starting at the salt seep and the last point starting at the freshwater area. In total, the whole transect was 16 meters. We collected 3 samples at each point for better distribution accuracy. And back in the lab, we sieved through and sorted through the samples. I used a dichotomous key to roughly identify the specimen and move the species into separated vials with 70% alcohol. I used the midge larva to complete genetic testing to identify the species. We extracted DNA from the species and ran gel electrophoresis to see if the DNA extraction was successful. If so, and bands were shown on the gel, I conducted a polymerase chain reaction, also known as a PCR, to amplify the DNA. If the PCR was successful, Dr. Cahill then sent the PCR to the Michigan State Genomics Core for sequencing. Once the sequences arrived, I used R, which is a programming language and software system for statistical computing and graphics, to create the phylogenetic trees. I used 8 Fangron and Sankner packages to use the phylogenetic code and use parsimony, NNI, and SPR methods for maximum accuracy. The trees I created used UPGMA, which stands for Unweighted Pair Group Methods with Arithmetic Averaging Algorithm, which basically means it's a rooted tree that assumes an equal rate of evolution, so the branch tips come to an equal stop. These pictures display a close phenotypic resemblance to the midges I found sorting through the sediment samples. The midges with the pink and greenish yellow hue are found in April and July, and the midges found with the clear or white color or found in July and October. So for the results, the line graph shows the distribution of biting midges at each transect point. The x-axis displays the transect point. Again, point 1 was the salt seep and point 7 was the freshwater area. The y-axis shows the species count. And as you can see, in April, most midges were found in transect point number 4. This was in between the salt seep and the freshwater. And in July, most midges were either located in transect point 3 or 5, which were either near the freshwater or near the salt seep. In October, had little to no midges. All midges were located in transect point number 1 at the salt seep. So for those unaware of what a phylogenetic tree is, it's basically a diagram that represents evolutionary relationships among organisms. So I would use the branching patterns in the tree to see how our specimen is related to the 2 identified species in the US. The phylogenetic tree I conducted combined Culicoides sonorensis and Culicoides varipennis genbank COI sequences in our own node sequence. So in this tree, you will see a distinct separate group of the Culicoides varipennis branches breaking off from the dominant tree. But then you can also see our own specimen labeled AEC14 in between multiple sonorensis and varipennis sequences. And if you look closely, you can see the 2 closest ancestors of our specimen is 2 sonorensis species labeled as KM989 and MF714. So in analyzing the results, we can infer that the distribution of midge larvae varies in each season. The majority of midges live in between the 2 extreme environments, the extreme rainy and high water levels in April, or the dry low water levels in July. There are multiple factors that can influence the midges to live in between the salt seep and the freshwater area. For example, salinity measures, nutrient and food distribution, and insufficient blood from livestock when the eggs are laid are prime factors in influencing the survival rate of the midges. So it could be that there wasn't enough of these survival factors in October's breeding season, since eggs are typically laid in winter. As for the phylogenetic tree, the unknown specimen collected from a transect will remain unknown. The results from the phylogenetic analysis come up inconclusive. So the results are inconclusive because if the species was truly either sonorensis or varipennis, it would be isolated from the other species. Results may be limited due to the sequences in GenBank that allows us to compare all three sequences, or since both species are closely related, it could be that the unknown specimen may be a C. varipennis complex or variant. So what's next? Late last year, I was successful in extracting DNA and conducting PCR in a couple of niches. So I will hopefully conduct more phylogenetic trees for accurate results in identifying the unknown midge species in the marsh. Also, there were a few limitations in conducting the distribution of the Culicoides larva in the transect since we only sorted through one repetitive sample in each season. So I would dig through and sort through more samples for better distribution accuracy. This is the end of the presentation, and I would like to acknowledge and thank everyone who made my research possible. I would like to thank Abigail Cahill for allowing me to be a part of the research team. Thanks to the research team for helping me with sampling and sorting through sediment samples. Big thanks to the biology department for funding the project, and Kurt Hellman for supplying and helping us to get to the marsh. Thanks to Chad Fadawa for the Michigan DNR permits and allowing us to go into the marsh. And finally, thanks to the Michigan State Genomics Corps for conducting the sequences Dr. Cahill submitted. Thank you, everyone, and enjoy the rest of your day.
Thank you all. My apologies for not having the video up. Dr. Cahill has mentioned in the chat that Rosemary is doing a community health internship with the Philadelphia Center. I wasn't able to be here today, but she may be able to answer questions if people have them. So again, if you would, please put the questions in the chat and we can ask those to Dr. Cahill.
So Doug White has asked, the two species seem to be separated on the cladogram. What's up?
That's a great question. So I don't know. And I will say also, all of the phylogenetic work was really Rosemary in terms of looking up what the different models are, trying different models, testing different models. She's presenting the combined tree today. So how separated those two species are depended on exactly what phylogenetics model she ran through the R programs. I think what's going on is that the C. varipennis is, we know it's a species complex, it's reported as a species complex, and so we know that there's cryptic species. We don't know that everything that's in GenBank labeled as C. varipennis is C. varipennis. I suspect it's not, and I suspect they're not each other's close, like that varipennis might not be a monophyletic group either, so they might not all be each other's closest relatives. So one of the things that we wanted to do, that Rosemary sort of started doing, and it was surprisingly difficult, was tracking down where all of the sequences in GenBank came from, and that information was really shockingly difficult to get. But I really want to know what species are most likely to be found near Michigan, because it came back as sonorensis, but we know that's a species that's primarily in the Western United States. So that's sort of what we think is going on.
And then there's a question for a non-natural science person. What is GenBank?
That's a good question. So GenBank is a basically a reference database of all molecular sequences generated through biology. I actually don't know how old it is or how long the mandate is, but basically, if you want to publish genetic data in scientific journals, this has been true for decades now, you have to make your sequences publicly available so that other researchers can access them. So it is a repository of sequences from different research projects, and it is I don't want to say there's no quality control, but we also know that there's stuff in there, for instance, that's not accurate, or maybe things that were entered 20 years ago that, new research has revealed that maybe it's not the species that they say it is, and people don't necessarily go back and update their entries. So we know there's some error associated with it as well.
All right. Thank you, Dr. Cahill. Thank you for your work on this, and I hope you'll help us in extending a thank you to Rosemary too. It was an excellent presentation. We are going to shift into our next presenter here. Thank you all again, and for those of you who might be joining us new, welcome to the Elkin Isaac Students Research Symposium. Just a reminder, we will be recording this session, and we ask that you don't unmute, but rather if you have a question or comment, please put it in the chat, and we will ask the presenters during the Q&A after they've finished presenting. It is my pleasure to introduce Sage Jost. And her presentation, let me switch the slide here. Sage will be presenting, and her faculty supervisor is also Dr. Cahill.
Hello, my name is Sage Jost, and I'm going to be presenting on the invertebrate species morphology and analysis throughout different environments. And I would say a special thank you to Dr. Abigail Cahill and to everyone who is joining here today. Next slide, please. So for the introduction, I have a few different pictures. So on the left are pictures of the Maple River salt seep where we took samples. And there should be some text on this slide. If you could please click next. Thank you. So what I looked at is the morphological data, and it's very important to record the morphological data and the differences in species that inhabit different environments such as these. So this particular environment is in inland salt marsh, which is a rare environment in Michigan. When you think of Michigan, there's freshwater lakes all around us. So to have a salt marsh in the middle of Michigan is pretty rare. So this environment is hard for species to adapt to because the salinity is different than what they're used to. And recording the morphological data allows us to account for which invertebrates can survive and adapt to these different environments. Next slide, please. So we also took samples at Lake St. Clair. Lake St. Clair is a freshwater lake in Michigan. And the place that we took them, I have a map on in a few slides, but we took them at a more wetlandy area. And so what we were looking at for the hypothesis, or what I was looking at for the hypothesis, is since these environments are so close together, but so different, like a salt marsh and a freshwater lake in salt composition and water flow, what types of macroinvertebrate species would be there, and how would they vary from locations? Next slide, please. So to the left is the salt marsh. We took samples, or I took samples on the Maple River floodplains. And as you can see, we took samples in 4 different areas. So that's 1, 2, 3, and 4. And then the salt seep is actually across from 1. And I know Rosemary in the last presentation talked about the salt marsh a lot too. And to the right is where Lake St. Clair is, and we took samples there as well. It's by Detroit, and in the lower image on the right is where those samples were taken. Next slide, please. So as I just stated, I went out to the Maple River with a group of people in October of 2019. We took samples in the floodplain and in the river itself. And then once back to the lab, I used a dichotomous key to identify and count the different species. On the right is an image of the Maple River area. So basically we were trudging through what looked like essentially this all the way out into the floodplains. And a dichotomous key is a go-to, it's like an identification of species. So it starts off with one feature, and then you work your way down as you're looking at the species to narrow it down to a specific species. And there should be a little bit more text, or no? Okay. So for the methods, continued, I put some pictures on here. On the furthest left is a damselfly larvae that I used the dichotomous key to identify as Caloptigidae. And there's 4 species of this native to Michigan. And the middle and the left are what I would be sorting through. So these are examples of slides that I would look at under the microscope. So in the middle picture, that's from the Maple River. And as you can see, there are arthropods, there are isopods, and there's other damselflies. And sometimes I would find stoneflies as well. And on the right is an example of a sample from Lake St. Clair. And as you can see, it's a little more sandy. There are a lot more shells. So the samples did vary quite a bit from the different sites. Next slide, please. So what I did was I compared the 2 samples that we took along the transect of the Maple River and the Lake St. Clair Metro Park area. And the morphological data allowed me to determine the species abundance through the different environments. And then what I did was I analyzed the data that I collected. So I wrote down all the species that I saw and I could identify under the microscope. And I put those all in a spreadsheet and I ran them through R to run 3 different tests. So I did species diversity, species richness, and an ANOVA analysis to determine if our results were significant. And R is basically like a programming code program. Next slide, please. So for the first slide, this is the Shannon Diversity Index. So the higher the number, the more diverse the site was. So as you can see, the Maple River had a lot more higher diversity than the Lake St. Clair data. Next slide, please. And this is an NMDS chart. So this looks at community composition between the 2 different sites. So the closer together the dots are, like for example, there are 2 at the top of the Maple River sample that are pretty close together. That means those communities are closer related. And same goes for in the Lake St. Clair data. There are 3 at the bottom that are closer related, but there is no overlap, which means that the community compositions between the 2 sites are different. Next slide, please. So this is another NMDS plot. The species at the top are actually different in taxa, and then the species that are clumped in the middle were more common. So it was less common to find like juvenile insects that were unidentifiable or beetles, but it was more common to find like all the ones that were jumbled in the middle. And again, there is no overlap in these as well. Next slide, please. For the ANOVA analysis, I just wanted to remind everyone that a p-value less than 0.05 means it's significant, and our p-value was 0.0483. So our results were significant, and that means that there was significant diversity between the 2 different sites that I looked at, which was awesome to find. And the Lake St. Clair data actually had lower diversity than the Maple River. Next slide, please. So what's next? Some future questions would be, why are some species more adapt to live in both conditions? How do they adapt? What does the overlap of the species being in both sites mean? What does climate change do to all these organisms living in these environments? And our results mean that we can do more research to answer more questions about these environments, and that species can adapt to live in different conditions to an extent, and what would be that extent, and that I think more research can be done in that as well. Next slide, please. I would like to give a special thank you to Albion College Biology Department and Dr. Abigail Cahill, to Chad Fedwa at the Michigan DNR for the permits at the Maple River, to Dr. Adrian Vasquez for sampling at Lake St. Clair, and to my research team, Chris Breen, Rachel Stander, Mike Sargent for helping collect the samples, and the SRP program for helping me conduct this research. Thank you guys, and are there any questions?
Thank you, Sage. And just as a reminder, if anyone has questions for Sage, if you could post them into the chat, I will then read them off to her. Our first question is from Angela Myers asking, why do you think the Maple River has higher diversity?
It could be because of where we were sampling. So the place that we were sampling was a floodplain, so there could have been species from the river that had come into the floodplain, or it could have been species that were not from the river that are normally in the floodplain that were just found there as well.
Our next question is from Doug White asking, do the variable conditions at the seep support a wider range of species?
From what I've noticed, it depends. I think yes, I would say yes to that.
We have a question from Patrick Mayo saying, good work, Sage, and then asking how much work was required in getting the permit from Michigan DNR.
I'm not entirely sure. The biology department got the permit for us, thankfully.
And Dr. Cahill has posted an update to say that there was not much work and she needs to renew it again. We have a question from Shanti asking, was there a species that you observed in your samples that you were surprised to see?
Yes. I think in the maple, if I remember correctly, in one of the samples, I did find a freshwater leech. And that was like, oh my gosh. Whenever I find leeches, I'm like, ah. So that was kind of surprising and really cool.
I will ask a question. Did you notice any like just, I know you haven't looked at it formally, but did you notice kind of patterns in terms of which species tended to be in both sites?
A little bit, like there are shells in both sites, but they're different types of shells. So like not really patterns, but I guess, does that make sense?
Yep, thanks. And then we have a question from Joe Lee Cullen asking, were there any spatial trends within a site that you could identify?
Not that I believe of.
I think we probably have time for one more question, if there is one. All right, thank you, Sage.
Thank you so much for your presentation. Just so we give it a little bit of time, making sure we're right on time so people can get here. I'll delay for just a second, but thank you all again for joining us for the Elkin Isaac Research Symposium. Just a reminder, we will be recording these sessions, and we would ask that you not unmute your microphone, but if you have a question or comment, please put it into the chat so that we can ask our presenter at the end of their presentation. It will be my pleasure to present our next presenter in just a second, but again, I want to make sure we're waiting so everyone who is here, that wants to be here, is here. You know, there are other sessions going on at the same time. All right, thank you all for your patience. It's my pleasure to present Alexis Wilkerson and her research presentation. Her faculty sponsor is Dr. Olapode.
Okay, hi everyone, I'm Alexis, and I'm going to be talking about the seasonal comparison of bacterial occurrences and their hydrolytic enzyme activities among different soil types. Next slide, please. So just to start with a little background, the main goal of my project was to examine and compare bacterial occurrences and their respective hydrolytic enzyme activities in different soil types on a seasonal basis. These soils were collected from the White House Nature Center, and a combination of field and laboratory tests were done to enumerate the bacterial populations in the different soil samples. So starting off, my study was based on asking 3 different questions. The first one being, what are the differences in the characteristics in the different types of soil? In this case, I explored 3 different soil types, including loamy, sandy, and clayey. And I believe that if there were differences in their inherent characteristics like bacteria, there would be differences in other things, other biotic factors. So the second question I explored was, what are the differences in the abundance of bacteria in the different soil types? With this, I believe that if the bacterial numbers differed, then the way that they functioned on an enzymatic level would also vary. And then the next question I asked was, what were the differences in the hydrolytic enzyme activities in the different soil types? And then how does that differ on a seasonal basis, that being fall versus summer? Next slide, please. So starting off, this is a separation of soil into classes or groups. This was originally developed by the USDA. So the 3 sides of the textural triangle represent increasing or decreasing percentages of sand, silt, or clay particles. From the lower left corner to the top of the triangle, the percentage of clay increases. The percent silt is identified along the right side of the triangle. And then from the top of the triangle to the lower right, the percent silt increases. And then the bottom of the triangle identifies the percent sand. And then from the lower right corner to the lower left corner, the percent of sand increases. And then at the point at which these 3 lines intersect will define the soil's texture. And then the textural triangle can also be used by drawing lines from percentages of soil particles vertical to the side of the triangle until they intersect to tell you what or how to classify your soil. So these classifications are determined by water content, organic content, temperature, and pH, which are all known to vary. Next slide, please. So this is a picture of the soil types and then where I collected them from. So loam is usually composed of mostly sand, silt, and a smaller amount of clay. By weight, its mineral composition is about 40-20% concentration of sand, silt, clay. It's typically characterized as being loose, fertile, easy to work with. Usually this type of soil has organic matter, making it an environment that's usually likely to have bacteria present. So clayey soils are finely grained natural rocks or soils that combine clay minerals with traces of metal oxides and organic matter. These are mostly composed of minerals containing variable amounts of water trapped in the mineral structure. And then sand soils are granular materials that are made up of finely divided rock and mineral particles. They're usually finer than gravel and coarser than silt, and usually this type of soil contains more than 85% sand-sized particles by mass. Next slide, please. So I'll get into a little bit of enzyme background. So enzymes have a special pocket on their surface called an active site. The molecule, in this case, in my research, it was fluorescent diacetate, which is a cell permanent esterase substrate, and they react with the molecules that they react with fits right into the pocket. And then the molecule or substance that the enzyme reacts with is called the substrate. And then that reaction is going to take place between the enzyme and the substrate at the active site. And then after the reaction is complete, the new molecule or substance is going to be released by the enzyme, and that substance is the product. Next slide, please. So I hypothesized that the 3 soil types would differ in their soil moisture, organic content, and nutrient concentrations. And I expected that the differences would also harbor different diverse bacterial populations, and there would be a difference in enzymatic capabilities and activity. So again, the main goal of the study was to detect and measure these differences in the soil particles and bacterial occurrences and their hydrolytic enzymatic activities during this study season. So usually loamy soil has an organic matter content making an environment that's likely to have bacteria present. That's a little bit of backing for my hypothesis, whereas clay and sandy soils have less organic matter content. Next slide, please. So the first thing I did was an organic matter analysis. This procedure was used for the estimation of soil organic matter by the loss of weight in a sample heated at a temperature high enough to burn the organic matter, but not too high to decompose the carbonates. So the soil was dried at 105 degrees centigrade for 2 hours, and then that was to remove the initial moisture. And then the samples were weighed and heated again at 360 degrees, and then weighed once more after that. And then once that data was collected, the percentage of weight loss on ignition was calculated, and that will give you the organic matter content. And the first picture is just added 105 degrees, and then the next picture is at 360. It got so high that the glass broke. But next slide, please. So next, I did a viable CFU bacteria count. So a linear dilution was done with the soil, and then it was plated on TSA, and I allowed the plates to incubate overnight. And then these are the results after about 24 hours. The first picture is the loamy sample, the second being sandy sample. And then loamy and sandy both have a great amount of colony forming units. And the third is the clay soil, which has a noticeably lower amount of colony forming units on it. Next slide, please. So the next thing I did was DAPI staining. So total bacteria numbers in the soil samples were determined using DAPI staining, which is a fluorescent stain that binds to the nucleic acids of cells. Epifluorescent direct counting is the best method available to enumerate the bacterial environment samples. So I took a sample, weighed it, and then preserved it in buffer, and then the cells were stained with 200 microliters of DAPI for between 3 to 5 minutes. And then when viewing bacteria using a microscope, they're usually invisible using bright field microscopy. So an epifluorescent microscope was used, and I counted DAPI stained cells in 10 microscopic fields in triplicates of soil samples with a handheld counter. And then DAPI stained cells fluoresced blue, and they were about 1 micrometer in size. You can see some bright DAPI stained cells on that picture on the right. Next slide, please. So the next thing I did was an FDA hydrolysis assay. An FDA assay is pretty useful to measure the enzymatic activity in extracellular enzymes like esterases, lipases, and proteases, which are all hydrolase enzymes. Microbes hydrolyze them in the presence of water, and they produce fluorescin. So in the FDA assay, I mixed about 2 grams of each soil sample with about 1 ml of FDA substrate and about 100 milliliters of buffer in a flask. And then those flasks were incubated in a shaker over the course of 24 hours. And then I checked the progress of the FDA after 2 hours, after 4 hours, 6 hours, 8 hours, and finally at the 24th hour. And then after each interval, the samples were spun down to separate the aqueous and organic phases. And I took the supernatant and placed 6 mils of it into a cuvette. And an additional cuvette served as a control, and that had acetone in it. But the intensity of fluorescent coloration and thus the enzymatic activity were then measured using a spec machine. I think I set it to about 490 nanometers. So yeah, those pictures just show the process of it happening chemically. But we can go on to the next slide, please. So the intensity of the yellow-green color was indicative of the overall amount of enzymatic activity in the samples. And then the quantification of the enzyme activity was measured by assessing the intensity of the color using a spec machine at a wavelength of 490. So as it goes on from 2 to 4 to 6 hours, the intensity, it increases. It's kind of hard to see with the naked eye, but if you really look up close in person, you can see it. But we can go on to the next slide. And then we have 8 hours and 24 hours, which you can see a lot more intensity increasing, especially from 2 to 24. Next slide, please. So now getting into my results, I have a summer and fall soil analysis. The top one is summer, the bottom one is fall. But the organic matter content in the summer and loamy sample was about 30%, sandy 2.5%, clay 6.6%, and then measured in the fall, loamy had a pretty dramatic increase, coming in at 46%, and then sandy pretty much doubled, 5.5%, and then clay had just a little bit less at 2.8%. Next slide, please. So next I have a summer versus fall CFU count. I just want to put it out there that viable counts are the cells that are alive, whereas DAPI counts are the cells that are living or dead. So summer versus fall CFU, we have a higher, we can see higher in the summer than in the fall. Yeah, we can go on to the next slide, please. So next we have a summer versus fall DAPI count. Again, summer has a bit higher DAPI count overall than fall, except for in the clay sample. That can be for various reasons, but usually the trend I saw was that summer had a higher DAPI count. Next slide, please. So I created a standard curve that was used to convert the OD readings from the spec machine to the amount of fluorescent released over time. On the left is that standard curve, and on the right is the fluorescent released over time. And loamy has the most enzymatic activity. It's lined at the top. And then underneath is sandy, and then at the bottom is clay. Next slide, please. So same thing, but for the fall, we created a standard curve and then used that to convert the OD readings to fluorescent released over time. Loamy, again, had the most fluorescent released over time, then sandy, then clay trailing behind there. Next slide, please. So to really make sense of these results on a seasonal basis, we observed a trend of organic matter driving bacterial numbers in both fall and summer. And then these results show fall and summer season by soil type, loamy, sandy, and then clay. Next slide, please. And then we have the same thing here, organic matter driving DAPI count with the same seasonal thing, loamy, sandy, and then clay. So I'll go on to the next slide, please. So in conclusion, not all environments are the same in live microbial populations. More activity in loamy soil compared to sandy and clay soil, and then organic matter content in soil is driving the bacterial occurrences, and then bacterial numbers are also driving FDA. So I'm going to wrap up. Next slide, please. So I'd like to give a special thank you to Dr. O. I've really created a nice bond with him, and I've really gotten a lot of opportunities through this research, so thank you so much. FIRSCA for sponsoring my research and funding, Dave and Kurt for all my lab needs when Dr. O wasn't around, Jill and Michael for the coordination of the symposium in general, and then the bio department for all that they do, of course. And that is all, and I'll be happy to take any questions.
Thank you, Alexis. Our first, and just a reminder for anybody who's just joining us or has since the beginning has just joined us, if you have questions, please post them in the chat. We do have a question from Doug White asking, would fungal abundance or activity in soil types influence bacterial numbers or activity?
I'm sure it would in some way or another. So with the DAPI staining and the CFU, well, I'm pretty sure if I let the plates incubate for a long amount of time, or I plated them on a different type of broth, then the fungal populations would grow and influence data. But with the DAPI staining, I'm just staining like the nucleic acids of bacterial cells, not of like fungal cells. I don't know how that differs, but I'm pretty sure it will skew the data in some way or another, yeah.
Probably have time for one more question. So we have a question asking, where would you like to see this research go next?
Unfortunately, it's not going to go anywhere because I am graduating now. If it weren't for COVID, I would have sequenced the bacterial populations, the genome, and found out like what exactly I was looking at bacterial-wise. But I am graduating, so this is the end of my undergraduate research.
Thank you. Great job, Alexis.
Thank you so much.
Thank you, Alexis, for that great presentation. Welcome to everyone again. Again, just a reminder, if you're joining us, thank you for being here for this presentation as part of the Elkin Isaac Research Symposium. Just a reminder, we will be recording, and we ask that you keep your microphone muted. If you do have a question or comment, though, please put it in the chat, and we will ask those to the presenter in a Q&A session after they have finished presenting. It is with pleasure that I would like to present Hannah Erickson. Oh, I'm sorry, my apologies. Let me get to the right slide. Again, I'm presenting Hannah Erickson, and her faculty supervisor is Dr. Cervantes.
Hello, everyone. My name is Hannah Erickson, and my advisor is Dr. Cervantes. And this is my presentation on mapping the mating type recognition pathway of Tetrahymena thermophila. Next slide, please. So this is just a little background information on Tetrahymena. This is a picture that I took of some Tetrahymena cells under a microscope. They kind of look like little ovals, and they just sort of swim around in there. So they are single-celled. They live in fresh water, so ponds, lakes, streams, etc. And they can be found pretty much everywhere, even here in Albion. I know Dr. Cervantes has collected some samples from around the Nature Center. So they are also a very well-studied model organism, and model organisms are pretty much ones that just have been looked at for a long time. They're usually inexpensive, and they are used to examine different biological processes. So next slide, please. Tetrahymena have 7 different mating types, so more than your average organism. And their mating is based on non-self recognition. So the rules are that they can mate with any other cell that is not their mating type. So a 1 can mate with a 6, but a 4 cannot mate with a 4. Next slide, please. So now the big question for my research is, how do Tetrahymena do this? Click. So that is a very complex question with an answer that involves a lot of moving parts. So to rephrase this in a more specific context, we ask the question, what genes are involved? Next slide, please. So to answer that question, we do need a starting point, and that starting point is going to be 2 genes called MTA and MTB. And MTA and MTB are known to be needed for mating type determination, which is the process of how a Tetrahymena cell selects one of the 7 mating types. And it's very important to note here that mating type determination is different than mating type recognition. So determination is how a cell selects its own mating type, and then recognition is the process of, during mating, of how a cell tells that another cell is different or the same as its own mating type. So while these processes are different, it cannot be said that they are completely unrelated. So MTA and MTB were used as a starting point for this research. Next slide, please. So data was collected from 2 types of Tetrahymena cells. I'll explain in more detail how this data was collected and what exactly it is. But I had a wild type sample, which contained both MTA and MTB genes. And then I had a mating type deletion sample, also called the knockout sample, where MTA and MTB were knocked out, or this just means that they weren't there, they weren't functioning. So in addition to these 2 samples, data was collected from 2 different strains of Tetrahymena, and this was just done because sometimes individual strains can become mutated, and we just wanted to avoid any sources of potential error there. So next slide, please. So this is where my workflow begins. We have our central dogma of biology on the left here. So beginning with our DNA, that is then transcribed into RNA. And I want to point out a very important detail here, that not all DNA is transcribed equally. So one gene may be transcribed into 10 copies, and then another may be transcribed into 100 copies. So this idea is called differential expression. So after that, in our central dogma, DNA is translated into protein. So if you could click. So for this research, genetic data was taken from this RNA stage, and then the RNA was reverse transcribed into cDNA, and then cDNA is sent through Illumina sequencing, and Illumina sequencing produces reads, which are a digital version, essentially, of the genetic sequence that we got from that original RNA. So next slide, please. So now we have our reads, that genetic sequence that we originally collected. And then I ran them through 3 main programs. The first one is called HiSat2, and this program aligns those reads to a genome that is pretty much just gives them a location on a tetrahymena genetic map that we have. And then the next program, HT-seq, counts those reads at each location. And then the final program, EdgeR, compares the number of counts at each location between those 2 samples, the wild type and the mating type deletion, and gives us our possible pathway proteins. So next slide, please. So EdgeR is a very important step in this workflow, and it pretty much measures and shows the genes that are differentially expressed, and this is just kind of a summary of how it does that. So we have our 2 samples, our wild type containing both MTA and MTB genes, and then the mating type deletion or the knockout sample with no MTA or MTB. And then EdgeR will compare the levels of gene expression, say for gene X in this example, and it does this for every gene in our data set. And then it produces a list of genes that have significant differences in the level of expression between those 2 samples. And we can attribute those differences to the effect of MTA and MTB because one sample has those genes, MTA and MTB, and one sample doesn't. So next slide, please. Between the 2 strains, I found 844 genes that were differentially expressed between the wild type and the mating type deletion samples. Additionally, there were 2,951 genes that were differentially expressed in the 1969 strain and 2,145 genes that were differentially expressed in the 210 strain. I did focus on those 844 genes because they were found in both strains, again, to just avoid any possible mutations or sources of error. So next slide, please. So the next step was to characterize those genes, and I did this using a website called Ghost Koala, and it gives you this nice colorful pie chart to look at. So of those 844 genes, 415 of them were downregulated, and downregulated genes are ones that have less expression in the mating type deletion sample, the knockout sample, compared to that original wild type sample. So the key categories of these results are the pink genetic information processing genes and the yellow environmental information processing gene sections. So next slide, please. The rest of the 844 genes were 429 upregulated genes, and upregulated genes are ones that have higher expression in the mating type deletion samples compared to the wild type sample. And we can see here again more of those pink genetic information processing genes and more yellow environmental information processing genes. These 2 categories are of particular interest for a few reasons. The first being that the mating process does involve a lot of genetic reshuffling. So it does make sense that MTA and MTB function may be connected to the processing of genetic information. And then the second one is that mating requires communication between and within other cells in their environment. So again, it makes sense that those environmental information processing genes are showing up here. Next slide, please. So moving forward with this research, we do want to explore and characterize genes from the environmental information processing and genetic information processing groups. In addition to that, I have found a lot of hypothetical proteins in this analysis, and hypothetical proteins are ones that don't have a name. There's not really much information known about them. So we do want to identify and spend a little bit more time looking into the significant ones that showed up in this research. Next, we would be able to use this research to create more knockouts. So other samples of tetrahymena where particular genes are removed, and then essentially this whole process is repeated using those knockouts. And then finally, all of these steps would just contribute more information to constructing the overall mating type recognition pathway. So next slide, please. I would like to say a big thank you to my advisor, Dr. Cervantes, along with the Albion College Biology Department, the FIRSCA program, and the Jane Seymour Killian Endowed Fellowship. Additionally, I would like to thank NCGAS, the National Center for Genome Analysis Support at Indiana University. They were able to provide us with server space and did a very good job of answering my many questions. So that is all I have. Thank you for listening. Questions?
Thank you, Hannah. Just for anybody who's just joined us, if you have questions for Hannah, if you could post them in the chat, I will go ahead and read them off to her so she can answer them. And while we're waiting, I'll start out with a question. Can you tell us how, so you mentioned you have a knockout that is missing those two mating type genes. How does one either create a knockout, or where does it come from that just has those specific genes missing?
Yes, it's not the easiest thing to create a knockout. The knockouts were created by Dr. Cervantes, and all of the data was collected before, so I didn't actually do that myself, but it is just a kind of, I don't know the exact details of it, to be honest. I know it can be difficult, though, and does take a lot of time and effort, so. I see.
Oh, we have a question from Abigail Cahill asking, what was the hardest part of the bioinformatics for you?
I would say the hardest part of the bioinformatics is there are a lot of things that seem very simple, kind of at the surface, but it is a very kind of nitty-gritty thing, and everything has to be very organized, and you do run into a lot of issues. So having optimism and a little bit of resilience is very helpful, and I have learned a lot over the course of doing my research, but it was a learning process for sure.
And we have another question from Dr. Cahill asking, did you or your collaborators have to build new pipelines for the tetrahymena? I know that genomes act very differently from other model organisms.
So our pipeline was, I don't want to say individualized, but we worked with NCGAS and kind of picked particular programs to use. So I know tetrahymena is a little bit finicky and definitely different, but I wouldn't say we had to make too many changes from a comparative pipeline. We're teaching, Dr. Cervantes is teaching a class now using a couple different programs that are also working with this data. So that's good as well.
Any other questions for Hannah? I'll ask another one. Do you, I don't know if you know the answer to this, but just all of the different mating types that exist, are there some that tend to pair up with each other more often than others, or is it completely random?
From what I know, it is completely random. They are just looking for anything that is not themselves. So I don't think there is any trends that go along with certain particular pairs.
And we have a question from Dale Kennedy saying, great job, first of all, and then asking what's next for you.
For me personally, I am done with this research, but it is kind of being continued by Dr. Cervantes and people in her lab, and I am taking a couple gap years to decide what's next for me.
Thank you, Hannah, for your presentation. We really appreciate it. Thank you all for asking questions. We are going to transition here into our next presentation. I'd like to remind everybody, several of you, many of you have probably heard me say this already, but thank you for joining us here for the Elkin Isaac Symposium. Just a reminder, we're asking everyone to keep their microphones muted, but if you have a question or a comment, even as we're going through the presentation, please put that in the chat box. We will ask the presenter in a Q&A session at the end of their presentation. We will read those questions to them. It is my pleasure to present, actually, I keep forgetting to do this, I'm sorry. Make sure I'm on the correct slides. It's my pleasure to present Rachel Stander. Her faculty sponsor is Dr. Cahill. Rachel, go ahead.
Thanks, Dr. Brown. It's great to see and hear from you again. Hi, everyone. My name is Rachel Stander, and I am currently a biology major, a psychology minor, and I am concentrating in secondary education. I'm actually currently student teaching, so I'm joining you all today from my biology classroom at my high school in Battle Creek, Michigan. And so today I'm going to be sharing a little bit about my research on the effects of road salt and its alternatives on freshwater invertebrates. And just like Professor Brown said, my faculty sponsor on this project is Dr. Abigail Cahill. Next slide, please. Okay, so the focus of my project was looking at road salt and really trying to understand some of its effects and impacts that it has on the environment and the ecosystem. And so first, in order to really understand that, we want to start looking at what the effects are that road salt has on water. And so road salt is primarily composed of a chemical compound as sodium chloride, which is abbreviated NaCl. And road salt is used as a de-icer on roads and sidewalks during the winter months in order to lower the freezing temperature of water. And so that's why we use this so abundantly, is it is actually able to lower the freezing temperature of water, which then quickens the rate of melting ice and snow. It is a really inexpensive method, and it's also really accessible, and a lot of townships and municipalities throughout the country use road salt as a de-icing agent. The quantity of road salt has increased tremendously in the past half century. It's increased tenfold, which for those of you guys that aren't science-minded, tenfold, an example of that is back in 1950, there was about 1 to 2 million tons of road salt administered on our roadways. And today there's about 10 to 20 million tons of road salt now administered. So it has increased tremendously in the last half century. So knowing that, what does that mean and how is it going to affect our ecosystem? Next slide, please. So what are the implications exactly of road salt? So the main implication is water toxicity of different freshwater systems. And so when road salt works its magic and it melts all the ice and the snow from the sidewalks and the roadways, it then drains into various different freshwater systems. And so it drains into our sewage systems, it drains into our rivers, which eventually makes its way into groundwater and then inevitably into lakes as well. And so we also know through previous research and literature is that salt pollution also has a harmful effect on both aquatic plants and animals, and it is incredibly toxic to said plants and animals. Additionally, it affects cars by causing salt corrosion, and it also affects our groundwater quality, which then can contaminate our drinking water potentially. And then lastly, road salt also increases the vulnerability of exploitation by invasive species in the environment. Next slide, please. So for my research, my big objective and really what I was interested in was examining different commercially available alternatives to road salt. And I was interested in how those alternatives affect the invertebrates, either similarly to road salts and just as harshly, or maybe there's some sort of alternative out there that is less toxic on these invertebrate species. And I focused on invertebrate species because most of Dr. Cahill's work is looking at invertebrates, so I was carrying on some of her work. And so in my study, I was really interested in seeing how these specific alternatives actually can alter and change the survival and potentially reproduction of these freshwater organisms. And so I hypothesized that alternatives will affect the invertebrates differently, and some will affect them more extreme than others. Next slide, please. So I'll begin by introducing what alternatives I selected to study in my experiment. So the first one is a beet juice ice melt. And I selected to choose beet juice because it's a safer alternative because the sugars that are encapsulated by beet juice are actually able to lower the melting point of ice. And this is also a fully biodegradable method. I also use sugarcane molasses, and sugarcane molasses works very similar to beet as far as a deicer, as the sugars in the sugarcane molasses is able to lower the melting temperature of water. The next step is pickle juice or pickle brine. For my experiment, I just used pickle juice, but a lot of municipalities do use pickle brine on their roads and sidewalks. And so this works by preventing ice from binding from the pavement, and then it also reduces the amount of chloride ions in the environment. And the chloride ions are really toxic to the invertebrates. And then lastly, sand. Sand is a pretty common method used to provide traction to roads and sidewalks. Though it does not actually melt ice or snow, it is an abrasive material that provides traction to our roads. And then for our study organisms, I chose 4 invertebrate species that are very common in the state of Michigan to experiment on. And so those included Daphnia pulex, ostracods, midge larvae, and then earthworms. And I've included pictures of those invertebrate species on the slide before you. And so it's important to note that 2 of my species, the ostracods and the midge larvae, were actually collected from the Maple River Salt Marsh, which is in Michigan. And this is kind of where my research career begun. I joined Dr. Cahill's lab my freshman year, and I was really interested in the research that she was starting to look at a salt marsh in the state of Michigan, which is in Maple River, and it is actually the only intact salt marsh in the state. And so from previous and preliminary research, we collected these 2 species of organisms. So these 2 species were already living in a naturally salty environment, and we were interested to see how these organisms would respond to the alternatives. Next slide, please. So for my methods, I exposed these different alternatives to the invertebrates. And so I created different concentrations of each of the alternatives. So based on the 4 that I used, so my sand, my molasses, beet, and pickle, I created a concentration and I exposed that to the invertebrate species. I also had road salt as a treatment just so I could compare and contrast how that would affect the invertebrates compared to the alternatives. And then of course I had a control and I used spring water, which I acquired from Victory Park. And so for my kind of setup, I placed the Daphnia, the ostracods, and the midge larvae in small glasses. And then for my earthworms, given that earthworms do not exist in an aquatic environment, they live in soil, I replicated that by filling many deli containers filled with soil, and so they lived in there. And so kind of the main difference between the worms and the other invertebrates is that for the worms, I had to actually spray the concentration onto the worms in the different cups. And so that's why you see those colorful spray bottles on the right-hand side of this right picture. Those are the spray bottles I use to expose the worms to the alternatives, whereas the other organisms were actually living constantly in the concentrations. Next slide, please. So continuing on with my methods, on a daily basis, I would come into the lab and I would collect data on the survival, how many organisms were living in each of my replicates, and also I was checking if reproduction had occurred yet. I would change the water about once a week because the water would get stale after a couple days. So I changed the water as well as fed all the invertebrates once a day. And then for my calculations, I opted and decided to calculate the integrated population density, which is using the trapezoid rule to calculate the area under the population growth trajectory. And so essentially, this method accommodated for the fact that data was not collected every single day. I was only able to collect data on the weekdays. On the weekends, I was off campus and unable to. So during this time, the integrated population density was the best method for calculations. Additionally, this method also allows us to account for asynchronous population changes across all the replicates and the fact that there are both increases and decreases occurring in all of the replicates. Next slide, please. So for my results, I'll start with my Daphnia. And the Daphnia, I have a picture included on the slide. It's just a little water flea, very tiny. You have to use a microscope to see all these invertebrates due to their small size. And so based on my experiment, I discovered that the Daphnia was strongly impacted by the pickle juice and the road salt. And so on the graph on the left, graph A, we see that A and B of the pickle and the salt on the lower, they reported very, very low survival. And so essentially the pickle juice and the road salt killed the Daphnia very quickly and shown to be very toxic to this organism. The beet salt and the molasses were kind of an intermediate. So the Daphnia did pretty well in the beet salt and molasses alternative, demonstrating that they could survive fairly well when exposed to that potential de-icer. Next slide, please. For my ostracods, I had kind of a similar result. And for the ostracods, they really were only impacted by the pickle juice. So of my alternatives, the pickle juice was again shown to be very harmful to the organism. Most of my ostracods perished when exposed to this alternative, and all the other alternatives, the ostracods did very well in. So we did not see any significant differences between any of the other alternatives on the ostracods. Next slide, please. And then last up is my worms. And my worms were actually not really impacted by any alternative. There wasn't any sort of significant difference between any of the alternatives for my worm species. And I'll explain why that is on my next slide. Next slide, please. Okay, so for my kind of take-home message on this whole project, essentially invertebrates perform better in different alternatives. So all of the invertebrates did better and responded better to different alternatives. So it's really hard to actually identify and find one specific alternative to road salt that doesn't negatively impact all of the invertebrate species. But overall, we see that beet, molasses, and sand are usually the least harmful of the alternatives, and generally the invertebrates that I experimented with did very well in that. The ostracods, which came from, again, they came from that salt marsh, and then my worms, which live in soil, so they were much less exposed to the concentrations compared to the other organisms, were less affected by salt. But overall, we see that high concentrations of road salt are pretty detrimental to population growth of invertebrates. So this work, the future, is continuing to search for de-icers that aren't toxic to freshwater ecosystems and limiting the amount of road salt that is notice my midge larvae not included in the results, and that is because there's a very small sample size, so I was unable to report that in the results. Next slide, please. Okay, lastly, just some acknowledgements. I'll wrap up here for you guys. So I would just like to take a big thank you to Kurt Hellman, who's the lab tech in the biology department, for all of his help throughout this project. My awesome lab partner, Chris Breen, for all of his assistance in my research, the Alabama College Biology Department for lab space, resources, everything that they provided me with. And then lastly, FIRSCA for funding this project, funding my summer FIRSCA, and all my research I've completed over the last couple of years, including Dr. Vanessa McCaffrey and Renee Krieger. Next slide, please. And then with that, that concludes my presentation, and I'd be happy to take any questions that you guys have.
Thanks, Rachel. If we have any questions for Rachel, if you could post them in the chat, I will go ahead and read those off to her. We have a question from Doug White saying, a few years ago, the college tried beet juice as a natural substitute for salt. There were negative human responses to the odor of the beet juice. Do you have a recommendation?
Yeah, when I was actually starting to do this project, a lot of faculty came up to me and were telling stories about the beet that they used in Albion, and they said it looked like blood was running through the streets because of the deep purple color that it gives off. But yes, regarding beet juice as a de-icer, it is a better alternative based on the research that I conducted. There is chloride ions in the beet, which is the really harmful component of the road salt, but at a far less concentration. So it does show to be a tad bit safer as opposed to the other alternatives.
Thank you, Rachel, so much for your presentation. Unfortunately, we have run out of time here, so perhaps you can answer a couple others in the chat. But thank you so much. Nice work. We are shifting here to our next presentation. For those of you who may be joining us, welcome to the Elkin Isaac Student Research Symposium. As a reminder, please keep your microphone muted, but if you have a question or a comment, please put it in the chat and we will read those to the presenter at the end of their presentation. So it's my pleasure to present Leah Serlin, whose faculty sponsor is Dr. Rebecca.
Hi everyone, my name is Leah Serlin, and today I am presenting my honors thesis, which was an examination of a multifaceted support system to provide stability for patients with arthritis while walking and standing in high heels. Next slide, please. So before we start, just a little bit about me. I'm actually named for Princess Leia. I'm a biology major in the honors program in the Wilson Institute with the goal of becoming a dentist. I transferred here from Oakland Community College. I'm from West Bloomfield, Michigan, and I am a cat and sheet cake enthusiast. Next slide, please. So how this project actually came to be. So the idea began when shopping for high heels. I was shopping for a wedding, and as a person with arthritis, I was frustrated because I couldn't find a pair of high heels that were functional and cute. So I had already actually begun a thesis, and I emailed my thesis advisor and said, can I change theses? Like, will you stay with me? And thankfully, Dr. Rebecca said yes, so thank you. Originally, the idea was to create a high heel with straps integrated into the design, but I decided the attachment was far more practical because it allows more than one shoe to be worn, and it also allows the wearer to choose their own shoe. So next slide. So this is just a little arthritis introduction. Rheumatoid arthritis and osteoarthritis are the two I'm going to go pretty in depth in today. So over 54 million people in the United States suffer from one of these forms of arthritis. Rheumatoid arthritis is characterized as an autoimmune disorder. So body is actually attacking healthy joint tissue. It also has morning stiffness, joint pain, and it's bilateral, meaning it's on both sides of the body. It's also an earlier onset. Osteoarthritis is a degenerative disorder, so it means that it's a wear and tear arthritis. So cartilage and connective tissue is breaking down, causing a bone-on-bone rubbing. And next slide, please. So here's some photos. On the left side, you can see the healthy joint tissue. In the middle, you can see that cartilage and connective tissue has been broken down, and there is that rubbing pattern in the middle there. And then you can see in the rheumatoid arthritis slide that the erosion has occurred as well. And the next slide. So some previous studies with arthritis and fashion, Chang et al. Found that many disabled people have a goal to blend in, and it causes them to sacrifice comfort for fashion. And this can become a real unhealthy obsession that they're constantly focused on, keeping themselves in trend as opposed to making sure that they don't have an issue with their comfort. Williams and Nestor found that comfort and style are the determining factors when diabetic and rheumatoid arthritis patients purchase shoes. And then in their study, they found that there was no satisfactory in both style and comfort when looking at the assistive devices, which were the shoes. Then DeBoer et al. Found that the major reason rheumatoid arthritis patients are not using their assistive devices, and in this case it was footwear, is due to the negative connotations of the style and appearance of the shoes. And the next slide, please. So other studies with arthritis and support systems. In the first one, it goes into how it's been shown that wearing a bracing device can decrease pain and activity limitations of wearers with osteoarthritis. Then in the second one, similarly, bracing and shoe orthotics and ankle support systems are all common additions to treatment plans for patients with rheumatoid arthritis because of these same ideals. Then the last one is not arthritis-based, but it's very important because bracing is used to prevent lateral ankle sprains in athletes. But it also gives a psychological benefit that the athlete is feeling more stabilized, and then the player is becoming more confident in their playing skills, and their quality of playing is actually increasing. All right, next slide. So the actual multifaceted support system, I actually have it here as well. So I'll be pointing out on here what's also on there. So the first one I'm going to talk about is the hybrid compression component, which is right here. So this is created with the idea of the hindfoot valgus joint, which is in the back of your ankle and your foot. It's affected by rheumatoid arthritis due to the shifting in weight distribution changes when you're having a flare-up versus when you're not. So your foot is constantly changing. So this can include pronation and supination of that actual valgus joint, causing the weight distribution in your midfoot to have problems and pain. So the compression ankle sleeve has boning incorporated on all four sides. So it's there, and then also just on either side here. And it maintains this neutral valgus joint. So it's not shifting at all. So your foot is in proper alignment, which alleviates a lot of stress. And it's also adjustable to fluctuations of ankle swelling. So if your ankle is more swollen, you can loosen it, or if it's less so, you can tighten it accordingly. Then the figure 8 strapping system, so you can see it across the top here, and that's also 2T in the picture on the page. It also goes underneath as well to really hold that foot in place. So the strapping system gives this reduced stress in the forefoot due to the increased stability of the unit movement. So this avoids joint overcompensation because your foot is now moving as a unit as opposed to moving in separate parts. And then finally, fashion elements. So it's fully adjustable and can cater to any look. You can change the colors, you can change the fabric, you can change anything you want about it. This one I actually have here has the rhinestones on it, just like the picture, and then lacing on here as well. It's kind of difficult to see because it's black. And then it is currently, all the designs I'm showing you today are patent pending. So there is that as well. And then next slide. So here are all the different views that I showed here, but in picture format, that's much easier to see, I'm sure. All right, and the next page. So the goal I set out to do with this thesis was to create something to help people in my position who want to wear a high heel, but just haven't been able to find the right one that gives the correct amount of support. And the hypothesis actually, after I created that, was then that this support system would provide the necessary support to make the high heel wearing experience more comfortable and stable for wearers with arthritis. So the next slide, please. So methods, so there's a couple of steps for this, but methods were that I had people fill out a Google form for consent and demographics prior to coming in for their appointment, also reminding them to bring a 1.5-inch high heel to testing with them. Then at the actual testing session, all three of the ones, the goniometer, dynamic gait index, and the Likert scale were completed twice, once without the attachment on, just with the regular high heels they brought with them, and then once with the attachment also on. So the goniometer measurements were to measure the alignment of the ankle and knee. I did it on both knees and both ankles. And then the dynamic gait index was 8 walking tests done to determine if the wearer is stable while walking. And each test was scored from 0 to 3, and then those scores were added to get a total score. The total possible score, the greatest score you can get, is a 24, and that is the most stable. And the least you could get is a 0, which would be the least stable. A score of 22 and above, so that 22 to 24 range, is considered to be stable with a very low risk of falling. And then finally, the Likert scale is a 1 to 7 scale done, where I asked 2 questions about how supported they felt in their high heels and how comfortable they felt in the high heels. And also when this was done the second time, I had an extra question asking how fashionable they believed that attachment was. All of this was statistically analyzed with t-tests to determine how significant it was. Next slide, please. So the demographics, you can see I had 31 participants, 8 with arthritis, 23 without arthritis. And then you can also see the mean age and standard deviation. And the next slide. So the results in this, the blue is unsupported and the pink is with the attachment. And then you can see arthritis, non-arthritis, and complete sample from left to right. Before I even tell you the numbers, you can see there's a drastic increase from without arthritis to, or not arthritis, sorry, without the support system to with the support system. So from left to right with the arthritis, it's a 12 out of 24 for without the attachment, 16 out of 24 for without the attachment for non-arthritis, and for the complete sample, it's 15 out of 24. And then from left to right again with the attachment on, the pink it is with arthritis, it was 23, non-arthritis was 23.4, and complete sample is 23.3. And the p-value for this was under 0.001, which was determined to be significant. And you can see the goal of the dynamic gait index was to get that over that 22 threshold, and it did do that. Then next slide. The goniometer measurements all went an average of 9.2 degrees towards proper alignment. And this indicates that the attachment does provide support towards better alignment when walking in the high heels. And it also was the p-value of under 0.001 as well. And the next slide. So the Likert scale on the left is how supported they felt without the attachment in blue and with the attachment in pink. And it was a 2.73 point increase. So it went from a 4 for without the attachment and to a 6.7. And then for comfort, it went from a 4 to a 6.6. And then this was also significant with a p-value of under 0.001. And then during this, anecdotally, subjects were asked how they felt in the attachment once they had it on and stood up, as well as when they began walking. And all the responses trended saying they felt more stable and much more comfortable, as well as confident in their walking while standing in those high heels. Next slide, please. So limitations, there are a couple things I should mention. Due to COVID-19, I did have to take out one of the tests I really wanted to do, which was the Neurocom Basic Balance Master System, to test the balance and get a really good graph of how people were standing in those high heels. Unfortunately, due to COVID, I was not able to bring people into the kinesiology lab, so this wasn't an option. And then also, I was the only one allowed in the room as the primary investigator when testing due to limiting the number of people involved. I would have liked to have an extra set of eyes. I also should mention this was my first time as the primary investigator. And then next slide, please. So next steps, after hearing feedback from participants, there seems to be a need for this type of product on the market. And other feedback also included creating a more fashion-forward design, which this one on this slide is that design I'm working on now. And then acknowledgements, thank you, Dr. Becker, for being the best advisor I could have asked for. Thank you, Dr. Cousins and Dr. Harris, for being on my committee. Thank you to the Honors Department and FERSCA for supporting me and funding me. And thank you to my friends and family for your support through this process. And a special thank you to Carter Fox for his support, as well as making my graphs on Python, because I do not know how to code. And then next slide for works cited. And then questions.
Thank you, Leah. If there are any questions for Leah, if you could post them in the chat, I will go ahead and read those off to her and for her to answer. So I will ask a question. Because I think it's really amazing that you've brought a new product that you're applying for patents for. What was that process like? Did you have a lot of background in building things? Like, how did you go about learning how to actually construct this?
Yeah, so there were about 7 prototypes before I got to this final one. I had some sewing background and I have a great needlepoint background, so I have some of that fine motor skill already, but once I actually got into it, it kind of got easier because I was just doing it daily. I got a great sewing machine. Highly recommend the Singer Heavy Duty if anybody is looking for one. Very easy to use, easy to like thread the needle and everything, not too many settings. So that's really how I went about doing it, was just kind of going for it and adjusting as needed. Yeah.
And we have a question from Dale Kennedy. She says, great job. And then also asks, are flat shoes a problem for women and men?
So with arthritis, I mean, it's just kind of any shoe's a problem. I can speak from firsthand for that. But flat shoes don't pose that inherent stability as much as, standing on this like tiny little peg of a heel. You can tip over pretty easily on that.
And we have time for one final question from Sean T. Brown. Were there any other products, fashion, or support that inspired you?
So I had looked at some designs of shoes that looked like they would be cute with the strapping, but they just weren't functional. So I kind of took that and went with, different designs I had seen and then tried to mold them into the strapping system that can still look fashion forward while providing that extra support.
Great, thank you so much, Leah. Thank you for an excellent presentation. We are going to transition here into our next presenter. For those of you who are joining us, welcome to the Elkin Student Research Symposium. Just as a reminder, we are recording this session, and we'd ask that you keep your microphone muted. But if you have any questions or comments, please put them in the chat, and we will read them to the presenter at the end of their presentation. It's my pleasure to present Victoria Carr. Her sponsors were Dr. Francis and Dr. Veet. Victoria, go ahead.
Thank you, Professor Brown. I'm Victoria Carr, and I'll be presenting on COVID-19 behaviors and grit. Next slide, please. So we're all familiar with the pandemic that is currently running rampant all over the world, with cases especially high in Michigan right now. So I promise I won't spend too much time talking about that. I know we're all talking about it all the time. But due to its highly infectious rates, a goal the CDC put in place last year was to control these infection rates in the hopes of flattening the curve or reducing the number of cases occurring simultaneously so as to not overwhelm hospitals and other healthcare centers. Places with especially high infection risk levels are places with close community living, such as residential colleges. In the efforts of keeping numbers low, many colleges have put in place safety guidelines for their students, such as mask wearing, staying in your dorm, not visiting other residential halls, and asking permission to leave campus. Next slide, please. When looking at rule-following behaviors, it's important to consider personality traits such as the Big Five personality traits and the Dark Triad. The Big Five personality traits consist of openness, or those inclined to be curious and imaginative, conscientiousness, or those with the tendency to be habitually careful and reliable, extroversion, or those who are outgoing and draw energy from others, agreeableness, or those marked by a tendency to be good-natured and courteous, and neuroticism, or those characterized by impulsivity and a tendency to be anxious and self-conscious. The Dark Triad consists of Machiavellianism, which is being manipulative and cynical, narcissism, which is being entitled and self-centered, and psychopathy, which is being socially callous and impulsive. Some personality traits that are often seen in those who follow rules are high levels of agreeableness, neuroticism, openness, and conscientiousness. On the other hand, personality traits that are often seen in those who break rules are high levels of narcissism, psychopathy, specifically traits such as shallow affect and lack of empathy, and extroversion. Next slide, please. In relation to personality, we see a strong relationship between individuals with highly open and conscientious personality traits and high levels of grit, which, if you remember, are two of the personality traits that are seen in people who follow rules. Grit, which is the main focus of my research, is defined as perseverance and passion for long-term goals during challenging times. There are two subskills that make up grit: gritty perseverance, which can be defined as putting continuous and strenuous effort towards achieving a goal despite hardship, and gritty passion, which can be defined as continuing towards a goal with unwavering enthusiasm over time. Next slide, please. Furthermore, grit has been associated with higher levels of well-being amongst college students and physician assistant graduate students. After controlling for the big 5 personality traits, previous studies found that high levels of grit predicted several things, including the completion of a selection course of Army Special Operation Forces, graduation from high school, continuation of an employee's work, and continuation of marriage. Next slide, please. Thus, we hypothesized that students with higher levels of grit would be more likely to follow safety guidelines in the fall 2020 semester than students with lower levels of grit because they were able to adjust when presented with obstacles. Next slide, please. For the current study, our participants consisted of a convenient sample of Albion College students recruited from the daily campus life email. In total, there were 156 participants, including 123 women, 29 men, 3 non-binary, and 1 gender fluid. Of that sample, 39 were first years, 30 were sophomores, 47 were juniors, and 41 were seniors. Next slide, please. Participants were first given a survey assessing their rule-following behaviors for the fall 2020 semester. This was a 19-item survey with Likert scale scoring. An example of an item was, I stayed within the Albion bubble unless given permission to leave. This survey is on a scale from 1 to 5, with 1 being strongly disagree and 5 being strongly agree. Participants were then asked to complete a survey measuring their grittiness. Participants rated how like them these items seem to be on a scale from 1 to 5, with 1 being very much like me, to 5 being not at all like me. This 12-item scale was broken down into the 2 subscales of grit, perseverance and passion. One of the gritty perseverance items was, I have overcome setbacks to conquer an important challenge, with lower scores indicating higher levels of gritty perseverance. While one of the gritty passion items was, I often set a goal but later choose to pursue a different one, with higher scoring indicating higher gritty passion. Next slide, please. A multiple linear regression was conducted to predict if COVID-19 rule following behaviors were based on students' self-reported gritty perseverance and gritty passion. Results found that the 2 predictors, gritty perseverance and passion, explained 7.2% of the differences in rule following behaviors. Next slide, please. As you can see here, both gritty perseverance and gritty passion significantly predicted rule following behaviors, but in different ways. Gritty perseverance, the blue line, increases as you move across the x-axis, showing more compliance with rule-following behaviors as gritty perseverance increases. While gritty passion, on the other hand, the green line, decreases as you move across the x-axis, showing less compliance with rule-following behaviors as gritty passion increases. Next slide, please. Results suggest that as gritty perseverance increased, so did compliance with the COVID-19 safety rules. However, as gritty passion increased, compliance with these same rules decreased. What this means is that while resilient students adjusted to these new conditions and were able to navigate the new rules, the most passionate students, on the other hand, were the most resistant to follow the new COVID-19 rules. Next slide, please. So what does this mean? Those who have higher levels of gritty perseverance will be able to adapt to new and difficult situations such as COVID-19, which begs the question, is it possible to train grit, and would training grit impact how students navigate new rules? It also means that those who have higher levels of gritty passion will resist these changes and rules, and it would be helpful to understand why and when these individuals were more likely to break the rules. Next slide, please. One thing that this study was unable to address was if COVID-19 behaviors impacted grit or if grit impacted COVID-19 behaviors. Examining this relationship could give us a better understanding of if the pandemic has had an effect on people's grit or if grit impacted people's COVID-19 experience. Another limitation is that this study was only conducted with Albion College students, and the safety guidelines at Albion were much different than many other colleges' guidelines. Next slide, please. For future directions, we would like to look into the two subscales of grit, perseverance and passion, more closely to see how they differ. Previous research has shown that these two subscales are in fact related, but are really two independent constructs instead of one. We would also like to look at how grit interacts with other personality variables, such as narcissism and extroversion, to predict rule-following behaviors in COVID-19. Previous research has looked into the role of personality traits and rule following on a larger scale, as we saw previous, but it would be interesting to see these same findings applied to COVID-19 rule following behaviors. Additionally, this summer with my faculty mentors, I will be replicating this study as a first summer project looking at a national sample. From this, we hope to be able to compare national results to Albion College students' results. Next slide, please. I would like to thank Dr. Andrea Francis and Dr. Marika Viet for all of their constant advice and mentorship throughout this project. I would also like to thank Shara Swan and the whole psychological science department for helping and supporting me along the way. Next slide, please. Thank you for everybody for coming to my presentation. If you have any questions, please let me know.
Thanks, Victoria. I will ask if anyone has questions for Victoria, if you can post them in the chat. We have our first one from Megan Webb. She says, great job, and then asks, can you explain the difference between grit and personality traits?
Yeah, there is some thoughts about this in a few different ways. Grit and conscientiousness are extremely related, and it's even been argued in some instances that they're so similar they shouldn't be considered different, which is one of the largest criticisms of grit. I'm not sure, there's not a ton of research looking at that, but there's just thoughts about that, which is something that's currently being looked into. At this point, I'm not entirely sure all of the differences, but they are incredibly related on a lot of levels.
Lindsay Moore asks, how concerned were you about response bias in your study?
Oh, we were definitely concerned about that, especially because these questions were pretty intrusive. We were asking students how they followed the rules and if they didn't. So there was concern about whether they were going to be truthful or biased or anything like that. We did our best to make it as easy for people to be truthful by not having names or emails connected to their actual responses, to try and separate any sort of personal feelings about your response because it wouldn't be tied to the participants in any way.
Any other questions for Victoria? I have one. I don't know if you know the answer to this or not, or I don't know if there's been work done on it, but is there anything known about, I just know there's been a lot of discussion about kind of the political dimensions of COVID rule following behavior. So I was wondering if there's any literature out there on that you know of on grit and like political orientation, like if that's something that's like an extra layer on top of COVID behaviors, or if there's already kind of an established relationship between grit and political affiliation that would just kind of feed into it.
I have no idea. I have seen some articles, not any journals, nothing peer reviewed, looking at that specifically during COVID. So there hasn't been any legitimate research that I've seen, but there's been some questioning of it. There was an article about following social distancing rules in a grocery store that was looking at grit, rule following, and political orientation. It didn't come to any specific conclusions, it was just speculating. I can imagine that there will be some sort of research on that in the future, but as of right now, I don't know of any.
And we have another question from Shanti Brown. Do you hypothesize changes in this data with less restrictive or more restrictive rules at other colleges?
I'm not sure. I'm interested. That's why I wanted to replicate this study with FIRSCA in the summer, because we're hoping to look at other college students. So I think there's potentially a chance, just because Albion is unique both in the rules that they put in place and the fact that it's a 4-year residential college. So I can imagine that there would be some differences there. I'm not entirely sure, but I can let you know at the end of the summer what we find.
And one final question. I likely missed this, but can you have both types of grittiness?
Absolutely. You can for sure have both. There is, arguments that they are two different variables, but are just, have a lot of overlap. But you definitely can have both. Some people have, higher one or the other. Some people have high both, low both. They're not, inversely related or anything like that. You can have both.
And did this have an impact on rule following if you have both?
I'm not sure. I can't remember if we saw that or not. I think that this was the most significant relationship that we found, was individually the two subscales. So I don't know if there was one that included both, if there's any type of relationship there. I don't know. Sorry.
Thank you so much, Victoria, for your work. I'll be excited to see you this summer and looking beyond. Yes, thank you again. Thank you everyone for the questions in the chat. We have one more presentation, but it is scheduled to start at 4 o'clock. So there is a 15-minute break here. So you're welcome to stay on this Google Meet and hang out with us, or join us back here again at 4 o'clock for the last presentation. Thank you all. All right, everybody. Can you hear me okay? It just gave me a note about my microphone. Is it working? Okay, I'm seeing nods, great. Thank you all for joining us. This will be the last presentation for Forum 4 of the Elkin Isaac Research Symposium. Thank you all for attending. Just as a reminder, for those of you who may be joining us, we are recording this session, and we are also asking everyone to keep their microphones muted. But if you have a question or a comment that comes up during the presentation, please put it in the chat and we will ask the presenters at the end of their presentation. It is my pleasure to present Erin Lewis and Lindsay Moore.
Hi guys, so my name is Lindsay and I am working on this project with Miss Erin Lewis. This project included a couple of other student researchers and is sponsored by Shanti Brown, Maggie Godfrey, and Barbara Keyes. This is our Albion COVID-19 vaccine needs assessment. Next slide, please. So today we will be discussing the background information on the COVID-19 vaccine, as well as the concept of vaccine hesitancy. We'll also be honing in on our current research project, as well as present what we have developed to be our educational website and what that looks like currently. Next slide. So a little background information, though the COVID-19 is still relatively new, there has been tremendous research on it thus far. Several studies have shown that in the United States, in the United Kingdom, and in Australia, the general public has many concerns about a possible vaccine. These concerns are specific based off of the group that you're in, and many at-risk and minority populations have uniquely specific concerns about the vaccine. The COVID Collaborative, which was created by the National NAACP as well as Harvard University, found that more than 50% of the surveyed Black community know somebody who has been diagnosed with COVID-19. However, less than 50% of the same community is willing to accept the COVID-19 vaccine. This brings us to the idea of vaccine hesitancy, which Erin will be discussing on the next slide, please.
So as Lindsay mentioned, there has been vaccine hesitancy that has been prevalent for the last couple decades, and especially now during COVID-19 and the vaccine rollout that we've seen within the last couple months. So the World Health Organization, or WHO, set out to define hesitancy and its scope, and to ultimately develop a model to categorize factors that influence behavioral decisions to accept a vaccine. So they gathered a group of SAGE members, which SAGE stands for Strategic Advisory Group of Experts, to find out what vaccine hesitancy's definition is and what it looks like within society. So what they found was, in the most basic sense, vaccine hesitancy is a delay in acceptance or a refusal of vaccination despite the availability of the vaccination. But what they also found was that vaccine hesitancy is very complex. It is context specific, meaning it varies across time, place, or location, and what specific vaccine is being given out. It is influenced by factors such as complacency, confidence, and convenience, which you can see in the Venn diagram figure. And I will come back to that and explain that after explaining what they found with scope. So in regards to the scope, again, they found that hesitancy is a behavioral phenomenon that is specific to context and the specific vaccine that's being given out. But they found that they measured it against the expectation of reaching a specific vaccination coverage goal. So that's what they compared it to in regards to the scope that they found. So to talk a little bit about the diagram, the Venn diagram with complacency, confidence, and convenience, it's called the 3Cs model, and it's a notable hesitancy model that gives insight to the complexity of the determinants of vaccine hesitancy, as well as the barriers for the lack of demand or increase of demand. So to explain it a little bit, we have confidence, and confidence is defined as in trust in the effectiveness of the safety of the vaccines, the system that delivers them, whether that is with the reliability or the competence of the healthcare workers or services that are administering the vaccine. and the motivations of the creation of the vaccine, or the policymakers who are creating the vaccine. And then we'll take a look at complacency. And complacency exists where perceived risks of the vaccine or the preventable disease that the vaccine is for has low risk. And they also believe that the vaccination is not deemed a necessary preventative action. So they don't, when people are complacent in that sense, they just do not believe that the vaccine is necessary. And then we'll look at convenience, and convenience has to do with whether that is physical availability, affordability, willingness to pay, or ability to pay, geographical accessibility, and ability to understand, whether that is through language or health literacy. And so as we can see, these three things can exacerbate one another. So for example, if we have low confidence or trust in the healthcare system, as well as high complacency, where they don't think it is necessary, then we'll see a low demand for the vaccine that is being administered. And then also it could be beneficial in the sense that if we have high confidence and high convenience, and so trust in the system, as well as easy convenience and accessibility to the vaccination, then we'll see an uptake in the demand for the vaccine. So overall, the definition for the vaccine hesitancy is necessary to ensure that our immunization programs, our policymakers, clinicians, and researchers would have a consistent standard term to cover the broad range of factors that cause the low vaccination uptake, while ultimately excluding those not related to personal or community behavioral choices. Next slide, please.
So our overall goal was to identify concerns that the Albion community members have about the COVID-19 vaccine. We understand that each community will have unique concerns about the vaccine, and in order to address Albion's concerns more efficiently, we developed a needs assessment in relation with the Albion Healthcare Alliance, the Albion chapter of the NAACP, and the city manager of Albion, to sort of create some tailored educational materials through the information that we received. Next slide, please. So our research project was a needs assessment, which is a survey. The survey specifically is about 5 to 10 minutes, and it can be taken in multiple forms, like a pen and paper. It's a phone survey. It's also taken online. And it asks questions based off of the participants' specific demographics, the access that they've had to the vaccine, whether or not they've been vaccinated already, and where they've gotten their sources of information, whether that be like social media or certain news sites, as well as any concerns that they have about the vaccine. This includes any concerns that they have previously, as well as current concerns that they currently have. This research is specific for the city of Albion, as well as Albion and Sheridan townships, and only open to participants above the age of 18. We had a goal of a goal sample of about 200 participants, and we began this research in the beginning of March. This research is ongoing and will end on June 1st. The overall goal, of course, is to develop Albion-specific materials to be distributed to the area. Next slide.
So a part of our research and our initiative to increase accessibility to educational materials and also reliable information to the community of Albion, we created an educational website. It is called Albion COVID-19 Vaccine Support, and I will take you through the website pretty quickly so you guys can get an idea of the layout and the information that we included for the citizens of Albion. So as you can see, we have a table of contents, and the educational website includes Albion contact resources, vaccine appointment signups, COVID-19 vaccination information. Next slide, please. And some informational videos, as well as nearest testing locations. So we have a welcome blurb explaining what our initiative is and what is the purpose of the website. And then we also highlight again what information is included on the website. Next slide, please. So at the top, we include Albion contact resources, which can include some of the members of the Albion Healthcare Alliance, as well as reliable sources or community members who have information or have answers to a lot of the questions people may have. And then we go down and we have the vaccine appointment sign up. So we included Marshall, so Oakland Hospital, the Calhoun County Health Department, as well as Young's Hometown Pharmacy, which is right here in Albion. And there are links to each of those websites to get signed up for the vaccine. And then we have the COVID-19 vaccination information, which includes some slides and commonly asked questions, as well as their answers from the Michigan Health Department of Health and Human Services. So you can click through those on the slide, and there's a couple very specific questions that are unique, and then some more commonly asked questions and the answers to those. And then we have some dialogue on the right-hand side of just commonly asked questions that's written out, so easy to scroll through and see the answers to some of those questions. And then we have what phase we're in. Next slide, please. And then finally, for the vaccination informational video or information, we have the informational videos, which were provided by some Albion College students, and they're just answering a couple of commonly asked questions, and there are some in Spanish as well. And then finally, we have the nearest testing locations, and we have a location in Albion, which is Washington Gardner, Hemingson's Drugstore, which is in Marshall, and then 2 that are in Jackson, and the links to each of those websites in order to get signed up for testing. And then we have our QR code and a small paragraph explaining our survey, and if anyone wanted to participate in the survey, that is how to access it. Next slide, please. So finally, here's a link to the website if you would like to go check it out. It is live, and we are updating it with new information as it comes. Again, this is for the community of Albion to be able to have reliable information, as well as access to reliable healthcare information and education. So that completes our presentation. I would like to thank our community partners, Albion Healthcare Alliance, the Albion chapter of the NAACP, the Albion City Manager, the Battle Creek Community Foundation for supporting this research project, and to our local pharmacies and businesses for allowing us to share our cards, information, and surveys at their stores and storefronts. And then lastly, a huge thank you to our faculty sponsors, Shanti Brown, Barbara Keys, and Maggie Godfrey for the support as well. Thank you.
If anyone has questions for Lindsay and Aaron, if you could please post them in the chat, I will read them off to them and let them answer. So we have a question from Megan Webb, who says, thanks, Aaron and Lindsay. Have you seen any of the data that suggests that the biggest group of COVID-19 hesitancy is primarily among rural whites? How might this relate to your project?
Actually, I have read some data on that. It's mostly because of a concern that they have about whether or not it's been tested specifically. The majority of our information we were looking at was looking at minority populations, but I did read a couple of pieces of information that the rural right community is also very concerned about it. But the focus of the specific project was looking at a community that is much similar to the diversity that is found in Albion, so we didn't pay as much attention to that. However, it does really relate to our project, and that is one of the main groups that is actually very hesitant about the vaccine. So that's a good point. Thank you.
And we have a second question from Irene, who says, great presentation. Do you see any resistance from a certain group?
Definitely among the older population, and then there are a few minorities that we found that have been hesitant, which leads back to problems that we've seen in the healthcare system that may be systemic that certain minorities have faced, which creates the low confidence in the healthcare system and the reliability of it. We see that with the 3Cs model a little bit. We use that, and we can see that with low confidence, there's not much trust in the healthcare system. So there is some hesitancy with the vaccination. And as Lindsay mentioned, just they want to wait to see what happens and to other people who get it first, or they would like to wait. So.
All right, thank you, Erin and Lindsay. Nice work. Everyone, this concludes this forum of the Elkin Isaac presentations. I'm pretty sure most of the other forums are also completing at this time. Thank you all for joining us here today. There will be recordings of these and the other sessions. My understanding is they'll be posted in multiple sites, including the main site for this symposium, as well as the YouTube channel for the college. So keep an eye out for those if there are ones you have missed. Thank you all again. Have a great evening.